Fakultas Kedokteran Universitas Lampung berada di jantung kota Bandar Lampung. Berdiri sejak tahun 2002, Fakultas Kedokteran Universitas Lampung memiliki lulusan yang tersebar di seluruh Indonesia. Sejak tahun 2012, Fakultas Kedokteran Universitas Lampung telah menggunakan kurikulum berbasis kompetensi dengan pendekatan problem-based learning.
Salam kedokteran.
Saya kedokteran.
Saya.
Satu kedokteran.
Satu. Berdirinya Fakultas Kedokteran Universitas Lampung diawali pada tahun 2002 dengan diterimanya SK Penyelenggaraan Program Studi Pendidikan Dokter. Pada waktu itu, program studi pendidikan dokter masih berada di bawah Fakultas MIPA Universitas Lampung. Seiring dengan perkembangan, pada tahun 2011, Fakultas Kedokteran resmi menjadi Fakultas ke-8 di Universitas Lampung. Pada akhir tahun 2018 diterima SK Penyelenggaraan untuk program studi S1 Farmasi dan program studi S2 Magister Kesehatan Masyarakat. Sehingga saat ini Fakultas Kedokteran Universitas Lampung memiliki 4 program studi, yaitu program studi pendidikan dokter, program studi profesi dokter, program studi farmasi, dan program studi Magister Kesehatan Masyarakat. Mahasiswa program studi pendidikan dokter Fakultas Kedokteran Universitas Lampung pernah memperoleh nilai tertinggi UKM PPD periode November 2015. Capaian yang kedua, perwakilan mahasiswa Fakultas Kedokteran Universitas Lampung pada tahun 2016 berhasil terpilih sebagai mahasiswa berprestasi tingkat universitas dan mewakili Universitas Lampung untuk mengikuti pemilihan mahasiswa berprestasi tingkat nasional. Mahasiswa program studi pendidikan dokter Fakultas Kedokteran Universitas Lampung pernah memperoleh medali perunggu pada PIMNAS ke-31 tahun 2017. Kemudian, rata-rata first taker program studi pendidikan dokter Fakultas Kedokteran Unila yang lulus CPT UKM PPD dalam 3 tahun terakhir adalah 80%, dan rata-rata first taker PSPD FK Unila yang lulus OSCE UKM PPD dalam 3 tahun terakhir adalah 90%. Kemudian, keketatan ujian masuk calon mahasiswa untuk masuk menjadi mahasiswa di Fakultas Kedokteran Universitas Lampung adalah tertinggi dibandingkan dengan program studi di lingkungan Universitas Lampung. Merujuk pada capaian-capaian prestasi tersebut, maka dengan belajar di Fakultas Kedokteran Universitas Lampung, mahasiswa akan dapat mengembangkan kemampuannya dan menjadi lulusan yang memiliki daya saing.
Laboratorium Anatomi, Laboratorium Histologi dan Patologi Anatomi, Laboratorium Biokimia, Biomolekuler dan Fisiologi, Laboratorium Mikrobiologi dan Parasitologi, Laboratorium Farmakologi, Farmasi Kedokteran dan Patologi Klinik, Laboratorium Ilmu Kedokteran Komunitas, Laboratorium Keterampilan Klinik, Laboratorium Farmasi, Animal House, ruang CBT, ruang tutorial, ruang kuliah pakar, ruang telekonferensi, ruang baca, musola, dan embung. Dalam kegiatan akademik, Fakultas Kedokteran Universitas Lampung memiliki fasilitas rumah sakit pendidikan utama, yaitu Fakultas Kedokteran Universitas Lampung turut mengembangkan soft skill mahasiswa dengan unit kegiatan mahasiswa, antara lain Dewan Perwakilan Mahasiswa, Badan Eksekutif Mahasiswa Fakultas Kedokteran, Lunar, Forum Studi Islam Ibnu Sina, PMPATD Pakis.
Sebagai seorang alumni, saya adalah bagian dari Fakultas Kedokteran Universitas Lampung. Selain karena fasilitas yang lengkap, kami juga dididik oleh dosen-dosen yang berkompetensi, baik itu untuk preklinik maupun dokter-dokter spesialis. Selain kita diajarkan tentang ilmu kedokteran, kami juga diajarkan tentang bagaimana kami bersikap sebagai seorang dokter. Sebagai seorang dokter tidak hanya pintar dalam ilmu kedokteran saja, melainkan harus juga pintar secara attitude. Dan di Fakultas Kedokteran Unila, kami dididik untuk bagaimana bersikap dokter yang memiliki attitude yang baik dan juga berkompeten. Untuk itu, sebagai seorang alumni, saya bangga sekali dengan Fakultas Kedokteran Unila. Jayalah selalu Fakultas Kedokteran Universitas Lampung.
Mahasiswa Fakultas Kedokteran Universitas Lampung telah menorehkan banyak prestasi, baik dalam bidang akademik maupun di bidang non-akademik, dan juga banyak penyelenggaraan lomba baik di kancah nasional hingga ke kancah internasional.
Fakultas Kedokteran Universitas Lampung merupakan salah satu fakultas kedokteran terbaik di Indonesia. Bila kita lihat dari sarana dan prasarananya, itu sudah sangat menunjang segala proses pembelajaran mahasiswanya. Hingga saya yakin bahwa saya dan mahasiswa lainnya dapat menjadi dokter yang amanah, yang dapat meningkatkan kualitas kesehatan masyarakat Indonesia, khususnya di bidang kesehatan.
Lampung, telung bersuka-suka, laman ram tinggal gunung, sang miru wajurai, ino lambang sai agung, lapangan jamo-jamo, giat ngebangun bumi Lampung, sang miru wajurai, ino lambang sai agung, lapangan jamo-jamo, giat ngebangun bumi Lampung. Sanun gajah zaman how, Lampung radu dikenal, hasil no pilado, rebutan kaum modal, wawai pemandangan no, di pinggir teluk Lampung, telung bersuka-suka, laman ram tinggal gunung, sang miru wajurai, ino lambang sai agung, lapangan jamo-jamo, giat ngebangun bumi Lampung, sang miru wajurai, ino
nama kalian? Dengan desain. Cek, cek, izin, di sini nggak sih? Cek gini, cek, gimana? Terima suara di Fakultas Kedokteran?
Hello, good morning.
Cek, cek,
Cek, cek, cek.
cek.
Sini berada ya.
Coba. Ya, oke, Pak. Oke, Pak.
Dua, tiga.
Halo?
Assalamualaikum warahmatullahi wabarakatuh.
Masuk. Ini sudah mulai, Pak.
May peace be upon you.
Good morning, ladies and gentlemen.
Tabi pun. The Honorable Rector of Lampung University, Professor Dr. Karomani, Magister of Science.
The Honorable, the Dean of Faculty of Medicine, Lampung University, Dr. Diahulan Sumekar, SKM MKes.
The Honorable, all dear guests and all the participants.
Let us begin by giving you a warm welcome to the
Opening ceremony of the International Conference on Agromedicine and Medical Science 2020.
Let us introduce ourselves. My name is Taufik Hidayat,
And my name is Fasya Azara.
and we will be your master of ceremony today. And it's such an honor for us to welcome you here.
On this special event, we have several agendas as follow. First, opening by Master of Ceremony, and singing the national anthem.
Next, reporting speech by chairperson.
Welcoming speech by the Dean of Faculty of Medicine's University of Lampung.
Followed by the welcoming speech and opening by Dean of Faculty of Medicine, University of Lampung, and followed by symbolic opening of the International Conference on Agromedicine and Medical Science 2020 by Rector of University Lampung.
The next is prayer.
Next, we have traditional dance performance.
And profile video of Faculty of Medicine's University of Lampung.
Continued by seminar by our keynote speaker, Mr. Kholis Abdurrahim Auda, PhD.
Also seminar by our invited speaker, Professor Dr. Wasim Haider,
Dr. Rizaidi Azli Mukhtar.
and Dr. Intan Safinar binti Ismail.
Closing by Master of Ceremony.
Especially for the oral presentation participant, there will be a break first before starting to the next sessions.
And the last is oral presentation.
As the start of our event, let's sing the national anthem of Indonesia, Indonesia Raya. Ladies and gentlemen, please stand up.
Now we're moving on to the speech session that is going to be started with a reporting speech by chairperson.
Please welcome Dr. Evi Kurniati, SKET MSc.
Thank you, Dr. Evi Kurniawati, SKET MSc, for the speech.
Now we will have a welcoming speech from the Dean of Faculty of Medical University, Lampung University.
Please welcome Dr. Dia Wulan Sumeka Renggani Swardani, SKM MKes.
Thank you, Dr. Dia Wulan Sumeka Renggani Swardani, SKM MKes, for the speech. Finally, welcoming speech and opening by Director of Lampung University.
Please welcome Professor Dr. Karomani, Magister of Science.
Thank you, Professor Dr. Karomani, Magister of Science, for the speech. After the words, we would like to request from Professor Dr. Karomani to officially open the International Conference on Agromedicine and Medical Science 2020, followed by symbolizations opening by Rector of Lampung University, Professor Dr. Karomani, Magister of Science.
Mulai kan? Ini kan ini. Assalamualaikum warahmatullahi wabarakatuh. Assalamualaikum. Enggak. Tadi itu karena gue habis
Satu, tiga. Mulai dong. Assalamualaikum warahmatullahi wabarakatuh. May peace be upon you.
Good morning, ladies and gentlemen.
Tabipun!
Un.
The Honorable Rector of Lampung University, Professor Dr. Karomani, Magister of Science.
The Honorable, the Dean of Faculty of Medicine, Lampung University, Dr. Dia Wulan Sumekar, SKM, MKes.
The Honorable, all dear guests and all the participants.
Let us begin by giving you a warm welcome to the
Opening ceremony of the International Conference on Agromedicine and Medical Science 2020.
Let us introduce ourselves. My name is Taufik Hidayat,
And my name is Fasya Azara.
and we will be your master of ceremony today. And it's such an honor for us to welcome you here.
On this special event, we have several agendas as follow. First, opening by Master of Ceremony and singing the national anthem.
Reporting speech by chairperson,
Welcoming speech by the Dean of Faculty of Medicine, University of Lampung.
and followed by symbolic opening and international conference on Agromedicine and Medical Science 2020 by the Rector of University of Lampung.
And followed by prayers,
Traditional dance performance, and profile video of Faculty of Medicine Lampung University.
seminar by our keynote speaker, Mr. Holis Abdurrahim Auda, PhD,
Seminar by our invited speaker, Professor Dr. Wasim Haider,
Dr. Ruzaidi Azli Mohtar,
and Dr. Intan Safinar binti Ismail.
and closing by Master of Ceremony.
Especially for the oral presentation participant, there will be a break first before starting to the next session.
And the last is oral presentation.
As the start of our event, let's sing the national anthem of Indonesia, Indonesia Raya. Ladies and gentlemen, please stand up.
Ladies and gentlemen, you may be seated. Now we're moving on to the speech session that is going to be started with a reporting speech by the chairperson. Please welcome Dr. Evi Kurniawati, Esket MSc.
Assalamualaikum warahmatullahi wabarakatuh. Tabi pun, My sincerest thanks to the Honorable Rector, University of Lampung, Vice Rector, University of Lampung, Dean and Vice Deans, the Medical Faculty, University of Lampung, Deans and Vice Deans, the other faculty, University of Lampung, keynote and invited speaker from Indonesia or abroad. All the lecture of the Medical Faculty, University of Lampung, as well as all the participant and paper presenter. All praise be to Allah Subhanahu wa Ta'ala, because today we can meet face to face online, strengthening our bond in the seminar of the first international conference on agromedicine and medical science. The first ICAM is organized in a collaboration between Medical Faculty, University of Lampung, Indonesian Society for Biochemistry and Molecular Biology, Indonesian Society for Biochemistry and Biodiversity. The international seminar is being held in celebration of the 18th anniversary or Dies Natalis, the Medical Faculty of Unila. The other academic activities than this ICAM seminar were the presentation of the paper, which will be published in Scopus Index Journal, and the Stadium General held at the beginning of October. This ICAM seminar is participated by 1,190 paper presenters from Indonesia and abroad. The ICAM seminar and paper presentation will last for 2 days, today and tomorrow, October 22 and 23. Our deepest and sincerest gratitude goes out to our Honorable beloved Rector, Vice Rector, Dean, and Vice Dean of the Medical Faculty, coordinators, lecturer, staff, student of the Medical Faculty, and Goodwood, the EO team that contributes a great deal to the realization of this extraordinary, prestigious event. Please allow me to also extend the gratitude to the speaker, both keynote and invited speaker. Dr. Holis Abdurrahim Audah, President of Indonesian Society for Biochemistry and Biodiversity. Director of Research and Community Service, Swiss Germany University. Dr. Asep Sukohar, Vice Rector of General and Finance, University of Lampung. President of Indonesian Doctor Association in Lampung Regional. Prof. Wasen Haidir from Central Michigan University, USA. Prof. Intan Sapinar binti Ismail, Department of Chemistry, University Putra, Malaysia, UPM. Profesor Zeli Nurrahman, Executive Board, Indonesian Society for Biochemistry and Molecular Biology. Dr. Taweporam Gedaram, Center of Excellence for Life Science, Ministry of Research and Innovation, Thailand. Dr. Miroslava Risova, Czech Republic, Technical University of Liberec Center for Nanomaterial, Advanced Technology and Innovation. Dr. Ruzaidi Azli Muhammad Mohtar, Deputy Director for Academic Affairs, Biotechnology Research Institute, Malaysia, Sabah University, who have fulfilled our invitation to share knowledge, have discussion, and strengthen the bond with us. Well, that's all for my welcome speech. I hope you will all enjoy your time here. And please let me say, welcome to the seminar of the first ICAM and paper presentation. May this series of events be a source of a new knowledge and science which will be beneficial to everyone. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Evi Kurniawati, SKMSC, for the speech.
Now we will have a welcoming speech from the Dean of Faculty of Medicine, Lampung University. Please welcome Dr. Dia Hulan Sumekar Rengganis Wardani, SKM MKes.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Tabi pun. First of all, I'd like to welcome the Honorable Rector, Professor Dr. Karomani MSc, Vice Rectors, invited speakers, deans and vice deans, all invited stakeholders, all faculty members and staff, and of course, all of students. Alhamdulillahirabbil alamin. Let us praise and be grateful to Allah Subhanahu wa Ta'ala, without whose grace we will not be taking part in the 18th anniversary of the Medical Faculty of the University of Lampung. Please allow me to first extend my gratitude to all of the speakers who will present their knowledge on this occasion, along with the all organizing committee members of this outstanding event. Ladies and gentlemen, Alhamdulillah, this year, the Medical Faculty of Unila has been serving for 18 years. As a faculty, it is of course still very young. There are still numerous goals we are trying to achieve for the sake of this beloved faculty of ours. Currently, our faculty has had 4 study programs, which are Medical Doctor Education Program, Professional Medical Doctor Program, Pharmaceutical Undergraduate Program, and Master Program of Public Health. We are currently still preparing several new academic programs at the levels of undergraduate, postgraduate, medical specialist, and also PhD program. On this special occasion, I would also like to share happiness of all academic members of our faculty for the achievement of the A accreditation, which is the highest level of accreditation. This is certainly of absolute happiness and pride for us, but however, there are more significant goals have to be achieved in the future. The main theme of this year's anniversary is synergy and innovation for the successful and competitive medical faculty of Unila. This theme bears our hopes of establishing synergistic relationship with a wide range of stakeholders and keeping innovating so that the faculty will never stop being successful and forming its students into competitive graduates. There are a series of activities of this year's anniversary, including this international conference. This conference will be conducted for 2 days, October 22nd and 20, which are today and tomorrow. The main theme of this international conference is Agromedicine and Medical Science. It is in line with the vision of our faculty. Attended by international and national speakers, this international conference is expected to be a medium through which the sheer abundance of motivation and insight related to medical science in particular that will be extended to lecture, staff, and also students, so that they will also be eager to carry out our research as well as publish their prominent scientific publication. Once again, I would like to convey our appreciation to all of you and we apologize for any unnecessary and inconvenience during this event. Dan kami mohon Bapak Rektor nanti untuk berkenan memberikan sambutan sekaligus untuk secara resmi membuka acara conference pada pagi hari ini. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Diahulan Sumekar Rengganis Wardani, SKM MKes for the speech. Finally, welcoming speech and opening by Rector of Lampung University. Please welcome Professor Dr. Karomani, Magister of Science.
Assalamualaikum warahmatullahi wabarakatuh. Abi pun. Alhamdulillahirabbil alamin. Wassalatu wassalamu ala asrofil anbiya'i wal mursalin. Sayyidina wa maulana wa habibina wa syafi'ina Muhammadin wa ala alihi wa sahbihi ajma'in. Yang saya hormati Ketua Senat, para wakil rektor, para dekan dan wakil dekan, para kepala biro, para kepala lembaga, ibu pimpinan Dharma Wanita, ketua SPI, para ketua jurusan dan kepala bagian, para kepala UPT, para ketua program studi, para kepala subbagian, para kepala badan, para koordinator kegiatan Dies Natalis ke-18 Fakultas Kedokteran Universitas Lampung, para dosen dan tenaga pendidikan, para mahasiswa yang berbahagia, baik yang hadir secara langsung maupun secara daring, yang saya hormati. Pertama-tama, mari kita mengucapkan puji syukur kehadirat Allah Subhanahu wa Ta'ala, berkat taufik, rahmat dan hidayahnya kita semua dapat hadir memperingati Dies Natalis ke-18 Fakultas Kedokteran Universitas Lampung. Syukur alhamdulillah atas capaian dan prestasi yang diraih oleh Universitas Lampung dan Fakultas Kedokteran Unila sebagai lembaga, dewan dosen dengan karya akademis inovatifnya, dan mahasiswa dengan prestasinya. Berbagai prestasi tersebut telah membawa nama FK Unila lebih dikenal, baik di tingkat nasional dan bahkan internasional. Para hadirin yang saya hormati, sebelumnya kita patut mengucapkan syukur dan terima kasih serta bangga kepada para pendahulu kita karena sejak tahun 1965 para pendahulu kita tidak henti-hentinya memberikan kontribusi untuk kepentingan dan kemajuan Unila. Karena itu, mohon maaf saya meminta kepada para hadirin sekalian untuk khusus menyampaikan terima kasih dengan membacakan Al-Fatihah untuk para pendiri Unila. Al-Fatihah. Semoga mereka yang masih ada sehat walafiat, dan untuk yang telah mendahului kita, mereka diampuni dan diterima amal baiknya oleh Allah Subhanahu wa Ta'ala. Bapak dan Ibu, tentu tidak banyak yang ingin saya sampaikan. Tujuan Dies Natalis ini adalah satu, untuk memperingati hari jadi. Kemudian untuk menyelenggarakan kegiatan-kegiatan baik akademik maupun non-akademik, serta mewujudkan adanya hubungan yang serasi dan harmonis antara pimpinan dan seluruh sivitas akademika. Bapak, Ibu, dan seluruh sivitas akademika, saya atas nama pimpinan Universitas Lampung dan segenap jajarannya, saya sampaikan ucapan selamat atas perayaan Dies Natalis Fakultas Kedokteran yang ke-18. Semoga Fakultas Kedokteran Unila semakin maju dan berkembang, dan lulusannya semakin berperan penting di masyarakat. Saya juga mengucapkan selamat atas pencapaian yang baru saja didapat, yaitu sebagai Fakultas Kedokteran terakreditasi A Tepuk tangan dulu untuk semuanya. Tentu pencapaian tersebut menjadi hadiah yang spesial untuk kita semua di hari jadi ini. Saya harap pencapaian tersebut tidak membuat kita terlena, karena tentu masih banyak pekerjaan yang harus kita selesaikan. Seperti yang telah disampaikan oleh Ibu Dekan tadi, bahwa usia 18 tahun merupakan usia yang muda untuk sebuah fakultas. Pada usia 18 tahun ini, berbagai prestasi telah berhasil kita capai. Selain itu juga berbagai kondisi telah kita lewati, mulai dari fakultas terakreditasi pada tahun 2012 lalu, yang mungkin karena kurang pandai kita mengalami penurunan, dan akhirnya pada bulan ini kembali menyandang predikat A tersebut. Membuat saya optimis bahwa kita dapat terus maju untuk mencapai visi dan misi kita. Bapak, Ibu, dan seluruh sivitas akademika, saya ingin menyampaikan dan mengingatkan kembali visi dan misi Unila, yaitu menjadi perguruan tinggi 10 terbaik pada tahun 2025. Mudah-mudahan mimpi ini dapat kita capai sepanjang kita semua melaksanakan apa yang kita tagline-kan, berinovasi dan bersinergi demi negeri dengan sungguh-sungguh. Pada kesempatan ini juga saya sampaikan salah satu roadmap yang saya dan jajaran sudah siapkan, yaitu sistem pemberian remun yang berbasis pada pemeringkatan kinerja fakultas. Sistem ini dibentuk sebagai salah satu upaya kita mewujudkan misi Unila, yaitu menjadi 10 terbaik universitas di Indonesia. Melalui sistem ini, saya harapkan dapat menjadi dorongan untuk para sivitas akademika untuk meningkatkan kembali kinerjanya. Salah satunya adalah melalui publikasi artikel ilmiah untuk meningkatkan nilai SINTA dan H-indeksnya. Selanjutnya adalah saya ingin menyampaikan bahwa mungkin kondisi saat ini tampaknya kita sedang lemah di outcome dengan minimnya jurnal publikasi, paten, haki, dan kinerja penelitian. Sehingga saya menginstruksikan bahwa semua remun akan diarahkan ke hal-hal tersebut untuk meningkatkan jumlah penelitian, paten, haki, jurnal publikasi para dosen di fakultas. Dana anggaran akan kita fokuskan pada kepentingan tersebut. Salah satu bentuk nyatanya adalah upaya peningkatan sistem IT kita dengan pemberian dana pengembangan untuk menambah akses poin kita supaya para dosen menjadi nyaman untuk pengembangan dirinya. Selain itu, saya mengusulkan untuk meningkatkan reward terhadap publikasi jurnal, paten, haki, dan karya inovasi lainnya. Berkenaan dengan itu, rangkaian kegiatan Kongres Internasional ini sangat kami apresiasi karena diharapkan melalui kegiatan ini dapat meningkatkan kemampuan dan kemauan para dosen untuk mengembangkan inovasi-inovasinya, mendorong para mahasiswa untuk ikut terlibat dalam penulisan artikel ilmiah. Selain itu, sebagai perguruan tinggi, saya ingin Unila menjadi sebuah komunitas akademik di mana para dosen dan mahasiswa bersama-sama aktif dalam proses pembelajaran dan penelitian dalam rangka pencarian kebenaran, sehingga universitas ini akan menghasilkan lulusan yang berkualitas tinggi dan berkepribadian yang mulia, serta juga akan dihasilkan pemikiran dan publikasi yang berhasil memberikan pencerahan kepada masyarakat. Akhirnya, pada hari yang berbahagia ini, saya ucapkan banyak terima kasih kepada pimpinan fakultas, para dosen, dan para staf penunjang Fakultas Kedokteran yang telah bekerja keras dan saling bahu-membahu dalam memberikan pelayanan yang terbaik dalam pelaksanaan Tridharma perguruan tinggi di fakultas ini. Semoga semangat kebersamaan semakin terbangun di fakultas ini, sehingga kita bersama-sama menghasilkan karya-karya yang pantas dibanggakan dan mendorong bangsa ini ke arah kemajuan. Dengan ucapan Bismillahirrahmanirrahim, maka secara resmi acara ini saya buka. Demikian, wallahul muwaffiq ila aqwamit thoriq. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Dr. Karomani, MSc, for the speech.
Moving on from the official opening, The next session is prayer. To Dr. Shazili Mustafa, Esket Biomed. The time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Peace, mercy, and blessing of God. Innal hamdalillah nahmaduhu wa nasta'inuhu. Praise be to Allah. We thank Him and we seek His help. Man yahdihillah fala mudillalah. Whomsoever Allah guides will never be led astray. Wa man yudlillahu fala mudillalah. And whomever Allah leaves astray, no one can be guided. Wa asyhadu an la ilaha illallah wahdahu la syarikalah. I bear witness that there is none worthy of worship but Allah, alone, with no partners. Wa asyhadu anna Muhammadan abduhu wa rasuluh. And I bear witness that Muhammad is his slave and messenger. Rabbana taqabbal minna. Our Lord, accept this from us. Indeed, you are the hearing and the knowing. Innaka antas samiul alim. Rabbana waj'alna muslimina laka wa min dzurriyyatina ummatan musliman laka. Our Lord, O Allah, make us Muslim in submission to you, and from our descendant a Muslim nation in submission to you. Wa arina manasikana wa tub alaina, and show us our rights and accept our repentance. Innaka antat tawwabur rahim. Indeed, you are the accepting of repentance and the merciful. Rabbana la tu'akhidna in nasina aw akhtana. O Allah, our Lord, do not impose blame upon us if we have forgotten or erred. Rabbana wala tahmil alaina isran kama hamaltahu alal ladina min qablina. O Allah, our Lord, and lay not upon us a burden like that which you laid upon those before us. Rabbana wala tahmil alaina ma la taqata lana bih. O Allah, our Lord, and burden us not with that which we have no ability to bear it. Wa'fu anna waghfir lana, and pardon us and forgive us and have mercy upon us. Anta maulana fansurna alal qaumil kafirin. You are our protector. You are our protector, ya Allah. So give us victory over the disbelieving people. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Our Lord, O Allah, give us in this world that which is good, and in the hereafter that which is good, and protect us from the punishment of the fire. Subhanakallahumma wa bihamdika asyhadu an la ilaha illa anta astaghfiruka wa atubu ilaik. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you to Dr. Shazili Mustafa Esket Biomed for the prayers. And that was only the beginning. On this session, we are going to present our welcoming dance on video, Sige Pengunten.
And followed by the profile video of Faculty of Medicine, Lampung University. To the committee, please share the screen on.
Fakultas Kedokteran Universitas Lampung berada di jantung kota Bandar Lampung. Berdiri sejak tahun 2002, Fakultas Kedokteran Universitas Lampung memiliki lulusan yang tersebar di seluruh Indonesia. Sejak tahun 2012, Fakultas Kedokteran Universitas Lampung telah menggunakan kurikulum berbasis kompetensi dengan pendekatan problem-based learning.
Salam kedokteran.
Sai kedokteran.
Sai.
Satu kedokteran.
Satu. Berdirinya Fakultas Kedokteran Universitas Lampung diawali pada tahun 2002 dengan diterimanya SK Penyelenggaraan Program Studi Pendidikan Dokter. Pada waktu itu, program studi pendidikan dokter masih berada di bawah Fakultas MIPA Universitas Lampung. Seiring dengan perkembangan, pada tahun 2011, Fakultas Kedokteran resmi menjadi Fakultas ke-8 di Universitas Lampung. Pada akhir tahun 2018 diterima SK Penyelenggaraan untuk program studi S1 Farmasi dan program studi S2 Magister Kesehatan Masyarakat. Sehingga saat ini Fakultas Kedokteran Universitas Lampung memiliki 4 program studi, yaitu program studi pendidikan dokter, program studi profesi dokter, program studi farmasi, dan program studi Magister Kesehatan Masyarakat. Mahasiswa program studi pendidikan dokter Fakultas Kedokteran Universitas Lampung pernah memperoleh nilai tertinggi UKM PPD periode November 2015. Capaian yang kedua, perwakilan mahasiswa Fakultas Kedokteran Universitas Lampung pada tahun 2016 berhasil terpilih sebagai mahasiswa berprestasi tingkat universitas dan mewakili Universitas Lampung untuk mengikuti pemilihan mahasiswa berprestasi tingkat nasional. Mahasiswa program studi pendidikan dokter Fakultas Kedokteran Universitas Lampung pernah memperoleh medali perunggu pada PIMNAS ke-31 tahun 2017. Kemudian, rata-rata first taker program studi pendidikan dokter Fakultas Kedokteran Unila yang lulus di PT UKM PPD dalam 3 tahun terakhir adalah 80% dan rata-rata first taker PSPD FK Unila yang lulus OSCE UKM PPD dalam 3 tahun terakhir adalah 90%. Kemudian, keketatan ujian masuk calon mahasiswa untuk masuk menjadi mahasiswa di Fakultas Kedokteran Universitas Lampung adalah tertinggi dibandingkan dengan program studi di lingkungan Universitas Lampung. Merujuk pada capaian-capaian prestasi tersebut, maka dengan belajar di Fakultas Kedokteran Universitas Lampung, mahasiswa akan dapat mengembangkan kemampuannya dan menjadi lulusan yang memiliki daya saing.
Laboratorium Anatomi, Laboratorium Histologi dan Patologi Anatomi, Laboratorium Biokimia, Biomolekuler dan Fisiologi, Laboratorium Mikrobiologi dan Parasitologi, Laboratorium Farmakologi, Farmasi Kedokteran dan Patologi Klinik, Laboratorium Ilmu Kedokteran Komunitas, Laboratorium Keterampilan Klinik, Laboratorium Farmasi, Animal House, ruang CBT, ruang tutorial, ruang kuliah pakar, ruang telekonferensi, ruang baca, musola, dan embung. Dalam kegiatan akademik, Fakultas Kedokteran Universitas Lampung memiliki fasilitas rumah sakit pendidikan utama, yaitu Fakultas Kedokteran Universitas Lampung turut mengembangkan soft skill mahasiswa dengan unit kegiatan mahasiswa, antara lain Dewan Perwakilan Mahasiswa, Badan Eksekutif Mahasiswa Fakultas Kedokteran, Lunar, Forum Studi Islam Ibnu Sina, PMPATD Pakis.
Sebagai seorang alumni, saya adalah bagian dari Fakultas Kedokteran Universitas Lampung. Selain karena fasilitas yang lengkap, kami juga dididik oleh dosen-dosen yang berkompetensi, baik itu untuk preklinik maupun dokter-dokter spesialis. Selain kita diajarkan tentang ilmu kedokteran, kami juga diajarkan tentang bagaimana kami bersikap sebagai seorang dokter. Sebagai seorang dokter tidak hanya pintar dalam ilmu kedokteran saja, melainkan harus juga pintar secara attitude. Dan di Fakultas Kedokteran Unila, kami dididik untuk bagaimana bersikap dokter yang memiliki attitude yang baik dan juga berkompeten. Untuk itu, sebagai seorang alumni, saya bangga sekali dengan Fakultas Kedokteran Unila. Jayalah selalu Fakultas Kedokteran Universitas Lampung.
Mahasiswa Fakultas Kedokteran Universitas Lampung telah menorehkan banyak prestasi, baik dalam bidang akademik maupun di bidang non-akademik, dan juga banyak penyelenggaraan lomba baik di kancah nasional hingga kancah internasional.
Fakultas Kedokteran Universitas Lampung merupakan salah satu fakultas kedokteran terbaik di Indonesia. Bila kita lihat dari sarana dan prasarananya, itu sudah sangat menunjang segala proses pembelajaran mahasiswanya. Hingga saya yakin bahwa saya dan mahasiswa lainnya dapat menjadi dokter yang amanah, yang dapat meningkatkan kualitas kesehatan masyarakat Indonesia, khususnya di bidang kesehatan.
The next session is closing of the opening ceremony and photo sessions with the Dean of Faculty of Medicine, University of Lampung. And the committee, please take the picture.
Foto pertama adalah dekan dan beserta jajarannya. Selanjutnya dekan beserta jajaran.
Mohon maaf sebelumnya.
Moving right along to our main event, seminar by our special guest, by Mr. Holis Abdurrahim Audah, PhD.
And it will be moderated by a lecturer from Faculty of Medicine, Lampung University, Dr. Diana Mayasari Esket, MKK. But before we start these sessions, I'm going to read the profile from Dr. Diana Mayasari as our moderator. Her educational background: she was graduated from Faculty of Medicine, University of Lampung for her bachelor degree, and graduated from Master of Occupational Medicine from Universitas Indonesia. And for her career experience, she is lecturer from Faculty of Medicine, Lampung University since 2009, the head of Department of Community Medicine since 2017, and the champion of Indonesian Hydration Working Group from 2018 till now. So please welcome Dr. Diana Mayasari Esket, MKK.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Welcome to the World International Conference on Agromedicine and Medical Sciences 2020, hosted by Faculty of Medicine, University of Lampung. I am Diana Mayasari. Today I will host this session as the moderator. Before we start the seminar, I would like to inform to all participants about the rules of this seminar. The first is, all participants please kindly mute the microphone during the webinar. And the second is, if you have any question to the speaker, please write down your question on Q&A room, or you can raise your hand on Q&A session. And after that, please fill out the attendance list from the link that will be shared by the community at the chat room. Well, this is my pleasure to be the moderator for this session on the topics Indonesian mangrove and their medicinal potential and opportunities and challenges. So, ladies and gentlemen, may I introduce our keynote speaker, Mr. Holis Abdurrahim Audah, PhD. Good morning, Mr. Holis.
Good morning, Dr. Diana. Anybody can hear me?
Yes, I can hear you clearly. Allow me to introduce you by reading your brief curriculum vitae. Mr. Holis is now a Director of Research and Community Services at Swiss Germany University in Indonesia. Mr. Holis graduated with a Bachelor of Chemistry and Biochemistry from Bogor Agricultural University. He also got his Master of Molecular Biology from University of Malaya and got his PhD of Biochemistry from Auburn University, Alabama, USA. His research interests are drug discovery from Indonesian biodiversity, antibiotics resistance, protein biochemistry, structural biology, in bioinformatics, genomics, proteomics, biomolecular interaction, and infectious disease. Mr. Holis has so many publications from his research published in international reputable journals, and also has been invited as speakers in many conferences, both in Indonesia and abroad. He is also a chairman of Indonesian Society for Bioinformatics and Biodiversity from 2016 to present, and the executive member of Indonesian Society for Biochemistry and Molecular Biology from 2015 to present. A very impressive curriculum vitae, Mr. Collis.
Thank you.
So, ladies and gentlemen, please join me to welcome our keynote speaker, Mr. Collis Abdurrahim Audah, PhD. Good morning, Mr. Collis. How are you doing this morning?
Good morning, Dr. Diana. Thank you very much for having me here today.
Thank you.
This
is also a pleasure for us to have you this morning. It is
such
an honor for us to have you in this seminar and for me as the moderator. So, sir, I would like to inform you that we will have 30 minutes for your presentation, and after that, we will have 10 minutes for Q&A session. Can we start now, Mr. Collis?
Sure, yeah. Thank you very much, Dr. Diana. Thank you very much.
So please, Mr. Collis, the time is yours.
Thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah, wassalatu wassalamu ala rasulillah wa ba'd. Let's us all praise Allah the Almighty for this very special event that we can today. Please, I would like to share my own slide, please. This is not the one that I like to share. This is for my next presentation. Can someone allow me to share my screen, please?
I'm sorry, Mr. Collis. Let the committee will replace
Yeah. Okay, let me just start it by introduction. Let's praise Allah the Almighty for His blessing. So we all of us today can gather at this very special event, and thank you very much for having me today. Peace be upon the Prophet Muhammad SAW, his family and companion, and his followers to the end of the day of judgment. So first of all, I'd like to thank directors, University of Lampung, and also the vice rectors, the deans, Dr. Dia, especially the deans of Agromedicine, and also the organizing committee, Dr. Dafi, for having me today. So this is a great honor for me to be here today, and a great pleasure to share with you all our knowledge and experience throughout these 2 days from today to tomorrow. Dr. Diana has mentioned about my CV. Thank you very much. I think that's a little too much introducing myself, but thank you again. So the title of my talk today is actually not that one. So if can I just share it? Okay, thank you. Yeah. So this is my. I would like to share today my current research focus. Anybody can see my slides today right now?
Yes, the slide has appeared.
Okay, so I'm going to be talking about Indonesian mangroves and their medicinal potential, and there are some opportunities and challenges. Again, my name is Collis Abdurrahim Audah. I work as Director for Research and Community Service at Swiss German University, and also Senior Lecturer at Department of Biomedical Engineering. And I also serve as Founding President for the Indonesian Society for Bioinformatics and Biodiversity. It is again my great honor to be here to give my talk, and hopefully it will give something to all of us, and maybe somehow inspire some of you that have not known much about mangroves. As myself, I recently knew about mangrove, but it is a fascinating plant indeed, because by getting more and more know about mangrove, we know that mangrove has a very tremendous benefits, especially for medicines. And we are Indonesian is a tropical country that almost all the coastline has mangrove leaf on the coastline. So this is the outline of my, this is the journey of myself actually, just a brief, so to get to know me better. Yeah. Tidak kenal maka tak sayang gitu. So as Dr. Diana mentioned about my CV, I travel around the world pretty much, because after finishing my undergraduate in Bogor, I continued my master degree in Malaysia, and also I did my PhD in the US in biochemistry, and later on I continued my postdoctoral studies also in the US at Yale University School of Medicine. Then after US, I moved to Middle East to establish a new faculty of medicine there at College of Medicine at University of Hayal, Saudi Arabia. Then I moved back to Indonesia, and the last one I've been for about 5 years in Swiss German University, and I enjoy it very much. So please allow me to briefly introduce my university. It is a very small private university, but indeed STU is one of the first established international university in Indonesia, founded in the year 2000 through the collaboration between Indonesian, Germany, and Switzerland government. It is a non-profit foundation under Indonesian law. We have three faculties with 11 bachelor and 3 master study program. The lectures is English and German as second foreign language. Currently, we have about 180 full-time staff and 90 part-time staff. So that's about it about my university. And this is the outline of my talk today, that I would like to introduce some about our potential and also research priority in Indonesia and wood, natural products, drug discovery, facts, and current development. And then I'll introduce briefly about Indonesian mangrove and their medicinal potential. And then we move to development of Indonesian natural products library for drug discovery, which is INPEL, that I'm working on right now. And we will conclude with future direction and acknowledgement. So as we all know that Indonesia has great potential as a great nation, to be a great nation in terms of different parameters, such as population. We are now nearly 280 million people in Indonesia. So whatever we have will be marketable here, even without outside market. In terms of geographical location, we are in a great position where we are the sanctuary of different kinds of organisms, both in marine and terrestrial land. And also we are a very rich country in terms of natural resources, either living organisms or any other minerals and metals and so on. So with all this potential, with all these assets, we can be a great nation for sure. So let's talk about Indonesian biodiversity. What's so great about Indonesian biodiversity that make us Indonesia as one of the richest country in the world in terms of biodiversity? These are some facts about Indonesian biodiversity. We have about more than 300 ethnic groups. So you can imagine if we work on genome, for example, genetic studies, we know that there are so many things to explore among these 300 ethnic groups. And also in terms of biodiversity, such as birds, amphibians, mammals, reptiles, fish, and vascular plants, we are among top 10 of countries in the world in terms of biodiversity. In fact, for birds, for fish, we are #1 in the world. And even for plants, we have about 30,000 endemic Indonesian plants specific to Indonesia. So we are just one of the mega biodiverse countries in the world in terms of biodiversity. So this is a very great asset for us to have. It's all depend to us, all of us, how we would explore this in the future. So this is another fact that I would like to share with you, that currently there are about more than 50,000 species of plants in Indonesia. So we have about 13,480 islands, more than 30,000 endemic species, and we know that about 6,000 species should use for jamu. So I think I can assure you that almost each of us here in Indonesia have tasted jamu once in your lifetime. So talking about the fauna, we are 12% of the world's mammals, 16% of the world's reptiles and amphibians, 17% of the world's birds, and 25% of global fish population, and about 1,650 coral reef fish. That's only in the eastern part of Indonesia alone. So having said all this, we can see that we are a very, very rich country in the world in terms of biodiversity. So now let's talk about what is the research priority in Indonesia? I take this information from the Ministry of Research and Higher Education at that time. So if we see here that from this table, we see the long-term plan from 2015 until 2044, it is seen clearly here that the natural resources-based applied research is put in the first priority in our government research plans, meaning that the majority of our resources, such as funding and policy and strategies, has been put into this research. That's why no wonder if many of us, I think it is very suitable, appropriate fit with our theme today, that agromedicine and spotlight of medical science research. It means that we are on the right track when we talk about agromedicine. So you can imagine Indonesia is a very big country. We have around 4,000 universities. Yeah, some of them are public universities. There are about 372 public universities on government-owned universities, and about 400 major private universities out of 3,940 universities. And with so many collections of herb plants throughout the country, making natural product-based research in Indonesia is the top priority. So it is a very remarkable number, very promising, but at the same time, we need to be very serious about this, where and how we want to bring our country in the next few years in terms of natural product-based research. So I would like to share with you some facts about World Natural Products Drug Discovery. So please let me introduce about the basic concept in nature that I'm sure that all of us today already understand about the basic concept of antigen and antibody, venom and antidote. The question is, do this also apply to medicines? Well, I'm a believer, I'm a Muslim, so as the Prophet says, for every single disease, there is a cure to it. So I'm very convinced that there is always a medicine to any kind of disease, including what we are experiencing right now, the COVID-19. So it's just a matter of how we discover it, how we find it. It is already there. Yeah, we just need to find it where it is and how to get there. So we've been knowing about this natural product-based medicine. It's a long story in many countries in the world, including Indonesia, China, India, and etc. That our ancestors have been using traditional medicine for quite so long until now. So this is a very strong basis for us to explore more, discover more. Why did the people, our ancestors, have been using this medicine for so long? And we just need to find and somehow modernize the way of acquiring it, and the way of utilizing it, and the way of how we produce the medicines. So these are some facts. The natural products derived medicines, we know about quinones, aspirins, and all alkaloid-based medicines. So this is a very interesting fact that some of you have not known about, that in fact, the 50% of FDA-approved drugs, not FDA, but in also other similar agency throughout the world, there are more than 50% of drugs actually derived from natural products or their derivatives. In fact, it is more interesting when we talk about cancer drugs. So there are about 75% of cancer drugs nowadays. I will tell you later on why this happens. Why about more than 75% of anti-cancer drugs derived from natural products. I learned this from a literature review, from Newman and Craig's natural review from 1980 to 2018. And the way they discover is actually about 1 in 1,000 natural product extract is used as medicine, and it is more favorable than development using synthetic compounds. So this is the reason why there are so many drugs yeah, either general drugs or anti-cancer drugs derived from natural products-based medicine. In the world, yeah, in the United States, in Europe, and in many other countries, there are companies, yeah, if not many, but there are companies that their job is just about collecting extracts, collecting fractions, or collecting molecules. For example, Albany Molecular Research in the New York, United States, they collect about 300,000 fraction collections. So they sell this fraction to pharmaceutical companies such as Pfizer, GSK, and many other companies that are interested in specific type of drugs. Yeah. There is also National Cancer Institute. This is the one that I mentioned briefly just now. Why 75% of cancer drugs derive from natural products? So apparently, NCI, this is under NIH, National Institute of Health in the United States, they collect about 230,000 unique extracts from all over the world to be used or examined for experimental research and translation for cancer research. And there are many others, company in the Netherlands and also Green Pharma in Spain that have been in contact with us recently. So I took this paper just to convince you that about NCI, who have been collecting, yeah, about 230,000 unique extracts derived from plant, marine, and microbial organisms that have been collected from biodiverse regions throughout the world. So if you can imagine our country that I mentioned, very rich, very much rich in natural products, in biodiversity, we don't need the rest of the world. We can just collect everything we have from our natural resources. I'm sure, yeah, very much sure that we can collect even more than just 230,000 unique extracts. Then we can use this for any kind of purposes in terms of drug discovery. So let's talk about drug discovery from Indonesian biodiversity, what we have and what we can do by utilizing this Indonesian biodiversity. So as I mentioned briefly that we are one of the richest country in the world. We are one of mega biodiverse countries in the world in terms of biodiversity. Yeah, I can skip this because we already showed this before. So there are so many kind of research. There are so many kind of natural resources in terms of plants, marine biota, and so on that we can explore. One type of plants or organisms that can be explored, yeah, for drug discovery purpose is mangroves. Yeah, I put the title Indonesian mangroves because, of course, we just want to focus on mangroves that exist in Indonesia, although we share mangroves throughout the world, including Borneo, because we also have collaboration with Malaysia. So let me just briefly explain about in more detail about Indonesian mangroves. So why we choose mangroves, ladies and gentlemen? So I'm sure you are all familiar with this site. Yeah, it's a very beautiful site. We took it in the last few years. So why mangroves? Yeah, why I'm personally myself very interested in mangrove research. So we have about along roughly 90,000 kilometer coastline in Indonesia, and it is home of about 20 families of mangrove, with more than 100 species of mangrove and their associates. Indonesia has the largest mangrove forest, or about 23% of total world mangrove forest. So if you see the number, 23% of total world mangroves, this is so huge, very large. Yeah, mangrove and mangrove associates have diverse and highly potential bioactive compounds. Yeah, so I took this and I republished this paper, this particular picture in our book chapter in Springer Nature 2019 last year. So this is the distribution of mangrove in Indonesia. If you see the spot, all red, yeah, all are red. It means that it's very highly densely populated mangrove throughout Indonesian coastline. So if we all together, not only myself, but many of us involved in mangrove research, there will be a huge environmental, social, and economical impacts. Yeah, we promote awareness of conserving natural resources. Yeah, because so far mangrove has been studied for conservation purposes or environmental purposes, but not too many people or researchers involved in research for drug discovery from mangrove. In fact, mangrove also consists of many kind of microbes. Yeah, we can isolate microbes from out of mangroves, and they are a good producer of antibiotics. We can also promote awareness of protecting natural resources because we feel the necessity of protecting those mangroves because we know the value of the mangroves. And this is a very suitable with our topic today, agromedicines, where people can, let's say, culture, grow the different kinds of mangrove in different types of environment. It can also boost the economical contribution to society by engaging people, especially in rural area, through cultivation and sample collection. Because this is our own experience that people get benefit from our research. Yeah, one person can give certain type amount of money to the people by collecting, growing the samples. And if you can imagine, there are thousands or hundreds of thousands people doing the same thing. It can literally move the economic very fast. And also accelerating the production of drugs from indigenous natural resources, yeah, throughout the direction of the synthetic biology. Because this is one of the challenges when we talk about drug discovery from biodiversity. We have a very limited amount of plants. We have very limited amount of biota that we have also to preserve. So one direction is how we can synthesize all the indigenous natural resources by using synthetic biology. This is, of course, another topic that will then be discussed later on in another opportunity.
I'm sorry, Dr. Collis.
Yes.
You still have, your time is 10 minutes more.
Sure, thank you, ma'am. So by doing research in mangrove, we can develop so many methods, yeah, and also we can make some model for Indonesian natural product library. So one of the example is species identification. Sometimes we don't know for sure, yeah, what species we are collecting. People say this is species A, but when we do really species identification using right tools, yeah. For example, we send our sample to Herbarium LIPI. We might get different types of species of mangroves, so we have to be sure about our species. Yeah. So this is also one study that we can pursue from a plant taxonomy point of view. So this is the certificate of species identification. Yeah. And what we are doing is, so far we are sampling about 8 species of mangroves, and out of this we collect about 16 parts of plants, and by using 4 different sample solvents we collected 64 extracts. I'm just briefly mentioning about how big the data that we can generate out of this mangrove research, yeah, or in general biodiversity research. So these are just some collection of storage, sample preparation and extraction, how we collect samples, how we transport the samples from one place to another, how we store the samples when upon arrival, and so on and so forth. Because we know that plants, most plants can be easily oxidized. So this picture I would like to show you because this shows the beauty of mangroves. We can see different colors from different types of part of mangroves, which indicate different types of chemical constituents. These are actually the quite common interest of the mangrove studies. So this again showing how mangrove are very rich in phytochemical. Yeah, we have steroid here, flavonoid, saponin, and tannin. Now let's talk about the drug discovery itself from mangrove library as antibacterial and anti-cancer sources. as we have our research studies here. So these are just some bioassay antibacterial studies show that mangrove is effective for certain types of bacteria, yeah, either local isolate and in fact for different types of pathogens. Yeah, for example, this one, yeah, we can see is very quite high number of inhibition index, and this is actually applied to MRSA, methicillin resistant Staphylococcus aureus, yeah, from Sonata sesolitis, yes, showing a very good example of antibacterial. And these are some data for anti-cancer, which are showing a very good lethality anti-cancer activity, but safe for human being. Now let's talk about what we are currently doing, which is the development of Indonesian Natural Products Library for drug discovery. Actually, this is parallel to our research now that we are establishing a database for INPEL, yeah, Indonesian Natural Products Library. So let me just briefly introduce about what is INPEL. So we know that we are facing health issues from day to day. Even right now, we are still in the pandemic era, which are being heavy burden for all of us in many different ways. And additionally, we are also facing different threats from different types of other infectious disease. Yeah, WHO report every 20 years there are about 30 new diseases, and currently 2017 WHO declared about 12 bacteria for which antibiotics are urgently needed. And we have to come up with solution how we overcome this issue, yeah, the limited drug discovery. We know that drug discovery is a very tedious, lengthy, and expensive process. It can reach up to 12 to 15 years, and it can spend about $1 billion just to produce one single drug. So we have to come up with other solution. For Indonesia, in the context of Indonesia, how? Yeah, and actually it's already applied in many different countries, which is by developing Natural Products Library. What is Natural Products Library? Which is a collection of natural products containing active compounds used for screening process of target biology. This slide, the picture is actually owned by GSK. They have the facilities of high throughput screening technology where they can screen hundreds of thousands drug candidates in single day. Yeah, I believe that there is only one in Southeast Asia, which is in Thailand, because last year I've been invited by the Ministry of Technology Thailand, that they have this kind of facilities that we can use together. That's why I invite one speaker from Thailand that can give a better insight about their facilities. So what is INPEL? INPEL is repository linked databases of Indonesian biodiversity with known functions, specifically for drug discovery purpose. So I put a note here with known function. So basically, there are two main objectives for INPEL, databases and repository. Yeah, we link this database and physical repository. So the role of INPEL is a window to Indonesian-based biodiversity research, a platform that links the virtual and physical repository of Indonesian biodiversity. These are some important features of INPEL. We can collect databases in terms of species identification, geographical locations, biological assays for drug discovery in this case, and some method developments. We can also collect extracts, fraction, compounds, and other metabolites. So, as I mentioned earlier, that there will be a tremendous amount of data can be generated from this research that need to be stored, in the information system, so everybody can see it, everybody can use it, everybody can utilize it for different purposes, especially in terms of drug discovery. Yeah, you can use for antibiotic activity, anti-cancer, structural elucidation, and so on and so forth. So it is extremely important for Indonesia to construct this database.
I'm sorry, sir, your time is 2 minutes.
All right, thank you. I will finish in a few minutes, in 1 or 2 minutes, okay?
Okay.
Yes, so this is the appearance of our INPEL website. Yeah, it's still under construction, of course. We still need to complete. So this is the existing libraries database in Europe. This is NIH, belongs to NIH. This is belong to United States. In United States, you can see every single plant in every state. But if we ask Indonesia, can we do that? Not at this moment. But hopefully after Impel, we can do so. This is from Campbell, from European countries. They have about collection 1.9 million compounds. Yeah, and this is knapsack developed by Japanese researcher. Yeah, and in conclusion, that by using this extract library, nature product lab, we can generate so much data. Yeah, and so far we collect about 64 extracts. Yeah, from different species, and also we also establish Impel, and we also establish consortium. And through future direction, high throughput screening technology. Now, of course, all of this we have to do it together. Yeah, through consortium, we cannot do it by ourselves. And so far, we have established consortium, and this is the MOU signing, including from Unila. Yeah, and this is collaboration with SUMS Malaysia Sabah. Last year, we took this opportunity. Just give me 1 minute, moderator, to share our happiness here that we have this.
Okay, sir, please.
Yes. Just 1 minute. Yeah. Can you hear me? Can you hear me?
Yes, I can hear you. I'm sorry, Mr. Hollis. We can't hear the voice from the video.
Oh, you cannot?
We cannot hear.
Okay, just give me 1 second then. Maybe I have to share. Just give me.
Yeah.
I need to share again. Yeah. Sorry, I thought you.
Universiti Malaysia Sabah UMS dengan kerjasama Swiss Germany ESGU di Indonesia sedang menjalankan penyelidikan mengenai kaedah mengurangkan jangkitan penyakit tuberkulosis TB dalam usaha menangani peningkatan kritikal selain penularan penyakit TB di negeri ini. Kerjasama penyelidikan bernilai RM400,000 itu masing-masing mendapat pembiayaan grant sepadan UMS berjumlah RM200,000 dan RM200,000 daripada SGU bagi tempoh 2 tahun bermula tahun ini.
Menurut Pengarah Institut Penyelidikan Bioteknologi UMS, Dr. Zarina Amin, kerjasama ini hasil daripada pertemuan dalam Simposium Bioteknologi Antarabangsa Anjuran UMS tahun lalu.
Kami di UMS ada mempunyai makmal tahap 3 biokeselamatan tu kan, di mana kami boleh culture BSL-3 organisms yang macam TB tu dengan selamat. Inilah specialty of UMS. Kita sebenarnya perlu showcasekan yang UMS ni ada begitu banyak research facility dan research strength.
Beliau menyatakan perkara tersebut kepada pemberita selepas Simposium Bioteknologi Antarabangsa kali ke-6 Anjuran UMS dengan kerjasama Asian Federation of Biotechnology di Kota Kinabalu. Sementara itu, Timbalan Pengarah Kajian SGU Jakarta, Indonesia, Dr. Kholis Abdul Rahim Audah memberitahu kerjasama antara kedua-dua pihak dalam projek ini melibatkan hasil bumi yang terdapat di Borneo seperti bakau yang berpotensi menjadi sumber perubatan.
So far we are in the process of extracting as many as possible mangrove extracts, and later on we will test or screen all those extracts towards tuberculosis and also as well as dengue viruses.
Mengenai
itu share kami untuk kerjasama antara Universiti Sabah dengan Malaysia. Just sebagai perkenalan untuk MABI, saya adalah Ketua MABI. Bagi ibu yang for those who want to register as a MABI member, you can go to the website mabi.id, and this is the organization. And I would like to thank everybody here for this that have been working together with me, including from so many different institutions. Yeah, and thank you again for the opportunity. Thank you, Madam Moderator.
Okay.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Mr. Collis, for very interesting presentation. So, sir, here we have some question in Q&A box. We have 8 questions now. Maybe I will read some of them.
Okay.
If there are questions that we cannot answer live right now, maybe you don't mind to answer it by typing the answer on the question.
Okay.
The first question is from Tri Nuring Tias. Her question is, mangrove has a very unique microenvironmental. Currently, many bioprospecting interest research for endophyte microbe associated with plant. Could you explain more what research have been done on it?
Sure, yeah. There are some groups in Indonesia, for example, in University of Diponegoro, University Jenderal Sudirman, that are working on endophyte microbes, especially associated with mangrove. So if you would like to join, you can join with us, because we also have WhatsApp group for marine microbes. So there are some, Prof. Oki is one example of researcher who are focusing on marine microbes. So yes, there are people who working on this endophyte microbes associated with plants.
Okay.
Did I answer your question?
She also asked that can we also store information that or data
Yes.
on INPL?
Very good question. That is the main purpose of INPEL. So everybody can store, like if you know NCBI, that people around the world store their data into NCBI. But now instead of NCBI, you can store to us INPEL. So because we also working on this, so everybody, that is the main purpose of this INPEL. So we know what have been done by Indonesian researcher. So we can work together, collaborate, avoid overlapping and so on. So yes, the answer is yes.
Okay. The next question, is there any safety data on mangrove extract for tropical and oral medication, sir?
Well, we've been tested for so far, not into human yet, yeah, because we, you know, mangrove research is very new. I can declare that I'm maybe one of the earliest pioneer in research mangrove in medicines, but we've been tested into not human, but still only by animal. But you know that there are different types of mangroves, yeah, different types, from a very nice sweet mangrove until a very bitter and toxic mangrove. So we need to be very careful on it. So when we talk about human beings, so of course we need to go to more into clinical trials, yeah, uji klinis, yeah.
So there are still many opportunities to do the research on mangrove.
Yes.
The next question, sir, from Evan, Mr. Evan. His question is, as you are explained that there are a lot of species of mangrove in Indonesia, whether all can be used as medicine or just some mangrove that can be used as medicine?
Yeah, as I mentioned earlier that we know that there are hundreds of mangrove species, there are hundreds of mangrove associates. So some are based on local wisdom that people have been using this for medicines. That's we can ensure that it is safe, yeah, but we need to be careful that some mangrove are also very toxic, so we need to be careful on that, yeah. So yes, I can just answer it in general, but so far the one that we've been using is actually safe for human, yeah. So we can, because our research is based on local wisdom. So let me also answer the question of this, what is the characteristic of mangrove extracts? The characteristic is vary because we use different types of solvents. But of course, as plant in general, there are many hydrophobic compounds. Yeah, we can extract by using hexane or any other solvent that is not hydrophilic. Yeah, but also there are some hydrophilic compound as well. Yeah, like that can be extracted using water, yeah, and also using different types or intermediate, for example, ethyl acetate solvent, yeah, from different types, either from leaf, bark, or root, yeah.
Maybe the last question, Mr. Collis, from Muhammad Iqbal. Are there any potential of coumarin compounds against cancer? And which one is better as a medication if you use the natural product for therapy as a single compound or combining compound?
Well, we cannot really answer which one is better because it's all depend on how we examine the characteristic of the compound or extracts. But of course, when we talk about drugs, modern drugs, or commercially available drug, people tend to use single compound. And before go to that, we need to understand clearly about the mechanism, the drug mechanism. That is why it all has to go through clinical trial. Either phase 1, phase 2, and 3, and then released or approved by the FDA, or in this case is by BPOM. So it really depends. It really depends on the types of extract, the types of compound that we are using.
Okay, thank you very much, Mr. Collis, for your time for answering all the, but I'm sorry for all the participants that we cannot answer all the questions. Maybe Mr. Collis has time to answer it at the next time. Okay, so sir, our time is up. Thank you very much, Dr. Collis, for your interesting presentation. I hope this session would give a new knowledge or a point of view for all participants, especially about mangrove and its medicinal potential. We are very pleased to have you today, Mr. Collis.
Thank you, Ms.
And looking forward to see you again on the next occasion, maybe.
Thank you. Thank you very much for the opportunity. Assalamualaikum warahmatullahi wabarakatuh.
Okay, Waalaikumsalam warahmatullahi wabarakatuh. So, ladies and gentlemen, all the participants, I would like to thank you for all the participants of this seminar, and I hope you enjoy all the session. And we are looking forward to see you in the next time of our event. And on the behalf of our committee, I would like to apologize of any inconvenience during this seminar. And thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh. See you again, Dr. Collis. Thank you very much.
Thank you so much for our incredible speaker as well as our moderator. We already hear the conclusions of this first lecture from Dr. Diana. Next, we would like to take a picture. To the committee, please take the picture.
Bisa patahkan kakiku, tapi tidak mimpi-mimpiku.
Kau bisa lumpuhkan tanganku, tapi tidak mimpi-mimpiku.
Kau bisa merebut senyumku, tapi sungguh tak akan lama. Kau bisa merobek hatiku, tapi aku tahu obatnya. Manusia-manusia kuat, itu kita, jiwa-jiwa yang kuat. Itu kita, manusia-manusia kuat. Itu kita, jiwa-jiwa yang kuat. Itu kita. Kau bisa hitamkan putihku, kau takkan gelapkan apapun. Kau bisa runtuhkan jalanku, karena ku temukan jalan yang lain. Manusia-manusia kuat, itu kita, jiwa-jiwa yang kuat. Itu kita, manusia-manusia kuat. Itu kita, jiwa-jiwa yang kuat.
kita. Bila bukan kehendaknya, tidak satu pun jelasmu akan bawa bahaya. Manusia kuat, itu kita, jiwa-jiwa yang kuat. Itu kita manusia kuat, itu kita, jiwa-jiwa yang kuat. Itu kita manusia kuat, itu kita, jiwa-jiwa yang kuat. Itu kita manusia kuat, itu kita, jiwa-jiwa yang kuat. Itu kita. Tak bisa patahkan kakiku, patah tanganku, rebut senyumku, hilangkan putihnya hatiku, tapi tidak mimpi-mimpiku.
Ladies and gentlemen, we will begin to the next seminar by our invited speakers such as Professor Dr. Wasim Haider, Dr. Zaidi Azili Mukhtar, and Dr. Intan Safinar binti Ismail.
And it will be moderated by a lecturer from Faculty of Medicine, Lampung University, Dr. Rani Himayani, SPM. Before that, I will read the curriculum vitae of Dr. Rani. For the educational background, Dr. Rani Himayani finished her Bachelor of Medicine in Faculty of Medicine, University Lampung, followed with ophthalmology residency program in University of Pajajaran, and years later, joined the Reconstructions, Oculoplasty, and Oncology Fellowship in National Eye Center, Cicando, Bandung. Please welcome Dr. Rani.
Okay, thank you MC for introducing me. Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning all audience. Welcome to the second session in the first day of our international conference on agromedicine and medical science 2020. I will be serving as moderator today. And before we starting the session, I want to explain the rules in the session divided into two parts. The first part is presentation by invited speaker. There are 3 speakers, and the next part is continue with discussion with all speakers. So, if there are any questions from all the participants during this meeting, you can write it in Q&A box, and don't forget, write the name of speaker you want to ask. Okay, it's now my pleasure to introduce the first invited speaker, Associate Professor Dr. Wasim Haider. He is an Associate Professor of Engineering and Technology at Central Michigan University. He is graduated as Bachelor of Science at Metallurgical and Material Engineering from Middle East Technical University, Turkey, and he get Magister of Science degree at Materials Engineering from Tampere University of Technology, Finland. And he received his PhD degree at Mechanical Engineering from Florida International University and finished his postdoctoral Material Science and Engineering in April 2011 from the Pennsylvania State University, University Park. And he has published around 59 papers in Repute Journal and 14 proceedings about 53 conference presentation, 20 poster presentation, and he also get 2 patents at November 2017 about multi-modal hands-free, and in 2014 get a patent about a method of alloying and surface treatment of nitinol to minimize platelet adhesion on blood contacting device. Okay, so now as to start the first invited speaker, allow me to invite Professor Dr. Wasem Haider to join with us and to present his presentation with the topic current trend in the drug delivery and technology. It is such an honor to welcome you here in this event, Prof. To Professor Dr. Wasem Haider, the time is yours, please.
Hello, good morning, everyone. My name is Wasim Haider, and I'm Associate Professor in the School of Engineering and Technology at Central Michigan University. And today I will be talking on metallic additive manufacturing for biomedical applications. So before I get started, I would like to thank the organizing committee members for organizing this conference, and my special thanks goes to Dr. Khalis Abdul Rahim for inviting me to this conference. So thank you. So before I get started, I would like to give you a brief overview about the Central Michigan University. So Central Michigan University is located in the state of Michigan, which is right here. And then a little star is representing the city of Mount Pleasant. Although there are no mountains, but it's a very pleasant city. So Central Michigan University has 7 different colleges. So we have a College of Science and Engineering and also College of Medicine and Health Professionals. So we do have all different sorts of departments which offer different majors and minors, including biochemistry, engineering, marketing, medicine, nursing, sociology, biology, physiotherapy, and so on. So in short, we have over 200 majors and minors at Central Michigan University. Now, specifically about the College of Medicine is the newest addition to the university's academic community, and it's one of 154 medical schools in the United States. So CMU College of Medicine's research efforts encompass basic translational, clinical, and public health areas of research. Also, finally, so we have all the 4 different seasons in Mount Pleasant, and we have a very walkable campus, as you could see, beautiful summers and winter and springs on the top. And then, yes, we do have a beautiful winter. You can see, it's full with snow. And nowadays, we are going through the fall season. So, okay, let's go to my presentation now. So I'll give you a brief introduction about the additive manufacturing here. So additive manufacturing is also commonly known as 3D printing. It is a technique or a process of joining materials to make objects from 3D models, usually layer upon layer. A true juxtaposition, as we are adding materials bit by bit, in contrast to removing bit by bit, which is a subtractive manufacturing, to make a final product. So here in the schematic diagram, you would see, the first step is the generation of the 3D model using any CAD software, such as SolidWorks. And then a command is given to the 3D printer, and the raw material is fed to the 3D printer. And then using the heat source, there's a selective melting at the designated scan rate, and a few millimeters thick layer is formed. And once that layer is formed, the second layer of powder is applied on the first layer. And then again, with the source of heat, it is selectively melted. And again, this step continues again and again. And finally, we have either a semi or a full finished product. So here I have a quick slide showing the mechanism of additive manufacturing. As you could see that we are printing a part here. So this right here is basically a blade which helps in bringing the powders, making it a thin layer. And once the laser source selectively hits the powder, it basically melts the powder, and right away it would start solidifying. And once it does that, the blade will roll back, remove all the extra powders, and then the new layer of the powder will be formed on the first layer. And again, the laser would be selectively melting, and then solidification process would start. And again, this process would continue while this part is being made. You could see that it is kind of moving in the downward direction, right? And finally, we have the completed or the finished product here. So an additive manufacturing technology is basically a game changer. So here I have a slide showing you cost versus complexity. So now you could see that with the conventional methods, when the complexity slightly increases, the cost can go very high. However, we see that almost a linear relationship, that even if the complexity is very high, the cost does not exponentially increase. Some of the important features is that it has an ability to make a complex geometric designs. It is basically a demand-based manufacturing. It's a customized manufacturing, and obviously it's economically viable. So here is an example for the different biomedical implants we can make with additive manufacturing technologies. On the top right, you see the acetabular cups here we can make. So we have also other orthopedic implants like femoral stems. We have all sorts of stents here, which we can make. So basically it tells, the manufacturer or the medical device manufacturer a complete flexibility, and they can make a lot of the product at one time at a different conditions. It basically helps in terms of a design freedom, and also it enables not only the complex geometry but also the in terms of product customization, and obviously it's quite economically viable. So this is the 3D printer we use in our lab, which is from Renishaw, a UK-based company, and the system we have is AM250. So again, additive manufacturing is a serial production process, is substituting the conventional manufacturing routes, right? So we can make solid, cellular, porous, all sorts of structures using this 3D printers. The recent advancements, it eliminates the bands associated to this process and restricting its applications in the past. So here are some of the examples where the additive manufacturing is used for biomedical applications. So we are talking about, like pacemakers, stents, other cardiovascular implants, spinal implants, orthopedic implants, dental implants, facial implants, and so on and so forth. So additive manufacturing is finding its ways to all these different sorts of implants. My research is mainly focused on metallic implants, so I'll be focusing, two major applications here, mainly, a blood contacting applications, and then a dental application. So these are the two things which we'll be talking in today's presentation. So before I go further, I would like to just quickly tell you that there are three major classifications for additive manufacturing. So we have a laser beam melting, and the sub branches of laser beam melting could be selective laser melting, laser cooling, laser metal fusion, and direct metal laser sintering. Similarly, for the laser metal deposition, we have a direct metal deposition, laser engineering, engineered net shaping, and laser cladding. And finally, sometimes electron beam is used as a source heat, so we call it electron beam melting. So that can be the one of the type as well. So now there could be different types of processing parameters which we can play with in additive manufacturing. And some of these parameters are, laser power, scan speed, hatch distance, layer thickness, spot size, and so on. But I would just take one feature, which is a laser power, and try to explain you from the material science point of view how it would affect, the microstructure and then ultimately the properties of the parts. So here you could see that once you have a high laser power, right, on the left, you could see that there's a larger melt pool and it goes deeper into the layers. However, when we have a high cooling rate or when we have a low laser power, which basically gives you the small melt pool, and then it only is basically in the top maybe layer or two. So now with that being said, so that once you have a slow cooling rate, you would get, because once you have a slow cooling rate, it takes a lot of time, right, for the pool to get solidified. So once you have a slow cooling rate, you would get a large size grains. And once you have a high cooling rate, you would get a small size grain. And obviously now when the microstructure is going to be different, all the properties such as mechanical, optical, deteriorative properties, magnetic properties, and so on, all these different properties of materials are going to be different based on their microstructures. So let's move on to my research here. So in this research today, I'll be talking about two main types of materials. So one is additively manufactured 316L stainless steel, a very commonly used steel for biomedical applications, for cardiovascular stents, for orthopedic implants, and so on. And also a titanium alloy, specifically it's a titanium, aluminum, and vanadium alloy. And now as a control sample, we are using commercially available 316 stainless steel and a wrought, titanium, aluminum, vanadium alloy. So we're going to be trying to compare here the commercially available alloys or the wrought alloys with the additively manufactured alloys. So once we have those alloys, so here's the sample dimensions given here. So once the samples were made, we just simply do the grinding up to 1200 grit size, and then we do the cleaning with ultrasonication, and finally we just do the water rinsing before we start doing the tests. So the first thing is we were trying to make sure that the chemistry of the surface on the wrought and additively manufactured are same. So we did EDS and X-ray diffraction. So on the left you could see that there's a wrought and additively manufactured. You could see that the chemistries, the compositions are almost the same. Similarly, on the right, you would see the XRD patterns or diffractograms for the wrought, which is blue, and the additively manufactured, which is the red color. So you could see that it's pretty much the same, okay? Which tells you that the chemical composition for the wrought and additively manufactured are same. And now here is the major difference when we took these images using the scanning electron microscope. On the left, you're going to see wrought, where you could see big grain size, right? Large grains. And on the right, you see additively manufactured, micrograph, where you could see a very small cellular type of structures. And insets kind of explains you how those cellular small structures look like. Moreover, you see some directional solidification as well here, and then you also see some melt pool boundaries and then the remelted regions as well. So again, what we can say here is that with additive manufacturing, we get some special and a very different type of a microstructure than the wrought materials. So now we're going to be doing different types of characterizations. So again, also to do a little bit more detailed chemistry analysis, we have run some X-ray photoelectron spectroscopy, and then we have done electrochemical impedance spectroscopy in 3 different mediums. So we have taken human blood serum and a phosphate buffer saline and a 0.9 molar NaCl. So we have 3 different electrolytes where we have done electrochemical characterizations. And then finally, we did some cell culture work where we have a MC3 G3 osteoblast cells, and then we did some biocompatibility studies as well. So let's look at the X-ray photoelectron spectroscopy data. This is the detailed chemical analysis on the top surface of the specimens, which clearly shows that additively manufactured and the rot have very similar chemistry. So we wanted to make sure that the chemistry is the same, which we did prove with XPS, as you can see here. And now when we look at the electrochemical impedance spectroscopy, so you could see that the semicircles which we obtain in 3 different electrolytes in serum, PBS, and 0.9 molar NaCl, you could see that the red curves have a larger diameter, right, as compared to the blue curves. So which tells you that the charge transfer resistance, for the additively manufactured sample is higher than the raw samples. Similarly, now when we look at the cyclic polarization experiments, again in the same 3 mediums, so these little circles, the dotted circles are representing, so this little circle here is representing the breakdown potentials for the raw sample. And this circle is representing the breakdown potentials for the additively manufactured samples. So now you could see that the breakdown potentials of the thin passive oxide film on the top of the metal in all the 3 electrolytes for additively manufactured is much higher, which means that the film is much more stronger, it's adherent, and it's not easily damageable in the case of additively manufactured when compared with the raw counterparts. So here we have the cell proliferation experiments. So in order to investigate the in vitro biological response of the wrought and additively manufactured, stainless steel, cell proliferation and morphology of MC3 G3 pre-osteoblast cells were assessed after 24 and 48 hours incubation period. So just briefly here, that cell proliferation and spreading on the implant surface are the initial steps in the cascade of cell-implant interactions, right? And it predicts the comparable in vivo response of the implant in terms of biofunctionality. So the surface texture and topographical features of the implant materials are the critical factors which may affect the growth tendency of the cells and consequently describes the implant tissue interactions. So here in this figure, you see the cell proliferation after 24 and 48 hours. We took up pictures and displayed them here. And you could see that both materials supported cell spreading and the appearance of stellar morphology after 48 hours of incubation, indicating the cellular compatibility of both samples. Although both samples supported spreading of cytoplasmic extensions, cell numbers remain increased on the surface of additively manufactured 316 after 48 hours of incubation, suggesting the enhanced cell survival and pronounced osteoblastic differentiation. Finally, this behavior suggested the better growth rates and a superior cell attachment characteristics on the additively manufactured samples. So similarly, we have run the Western blots. So again, just to briefly tell you that the finest biocompatible materials not only support cell proliferation but also provide optimum environment for the specific genes in the cells that improve their ability to differentiate into osteoblastic, or in osteoblast cells. So we have taken, RunX2, osteocalcin, and cleavage caspase-3. So you could see that additively manufacturing is behaving better for osteocalcin and cleavage caspase-3. However, in the case of RunX2, by the way, RunX2 is an anti-proliferative gene that minimizes cell proliferation once pre-osteoblast cells start terminal differentiation. Therefore, we use this protein as a biomarker for immature osteoblasts. So to determine the effect of microstructure on differentiation, expression of RUNX2 were assessed by Western blot in MC3T3 cell cultured on the wrought and additively manufactured samples for 72 hours, by the way. So consistent with the study cell numbers after 24 hours and incubation of osteoblast differentiation, RUNX2 was equivalent in numbers cultured on either samples. So these data indicate that both materials supported differentiation of osteoblasts. And as explained earlier, the refined structure of AM samples most likely provided better surface for cell adhesion and migration by promoting the deposition of extracellular matrix and cells during the differentiation process. So thus the RUNX2 compared to the raw samples suggest better biocompatibility of the additively manufactured stainless steel samples. So finally, just to conclude this project, about that it is that fully austenitic stainless steel was fabricated using additively manufactured technology, and then heterogeneous microstructure containing 500 to about 800 nanometers subgranular structure was observed. Higher corrosion resistance was observed for additively manufactured samples in all the three different electrolytes. And finally, additively manufactured samples supports better cell proliferation and differentiation. And very quickly jumping on to my second and the last project is about titanium, aluminum, vanadium. So where we have used again the Renishaw AM250 system under these conditions here to print my samples. And again, the intention was to use, for the dental implants. So here's a setup explaining about the electrochemistry, or electrochemical testing. So here is our working electrode Ti64, which is, we have two types. We have a raw samples and additively manufactured samples. And then we used artificial saliva as an electrolyte medium. And then we use platinum as a counter electrode and a reference, standard reference calomel electrode as a reference electrode. So I know this is kind of crowded table, so I want you to start maybe on the right sides where it shows you that we have used artificial saliva as our electrolyte, and then this artificial saliva mainly has all these different compounds and their compositions are given, right? And now what we did was that we have added, so I have divided our electrolytes into two groups as a group A and group B. And finally, the table on the far right, for the far left, shows you the chemical composition or EDX analysis for the ROT and SLM, which kind of pretty much shows the almost similar compositions. So here is the microstructural examination and XRD analysis. So now you could see that when we look at the microstructure under SCM, we could clearly see that a thinner or a more columnar type of beta grains as compared in the SLM or in the additively manufactured as compared to the rod counterparts. So sometimes I would use this term selectively laser melted. It's the same as additively manufactured samples. Okay, so in this project I'm going to be using, instead of AM, I'm using SLM as a selectively laser melted samples. So again, when we look at the compositions using the X-ray diffraction, we could see pretty much the same thing, except that we saw low percentages of beta in the SLM as compared to the rod counterparts. And this is also shown here in this table, the compositions for alpha, alpha prime, and also for the beta. Here is the electrochemical impedance spectroscopy analysis. Again, this is for group A and this is for the group B. So now you could see that in the group A, almost on the first three samples, you could see the better or the higher semicircle for the SLM or the additively manufactured samples, which is the red curve as compared to the blue curve, which shows that it's behaving better. Except that in in artificial saliva and the in artificial saliva in this one basically, and the lactic acid solution. However, when we look also at on the group B, you could also pretty much see that for most of the solutions additively manufactured is behaving better when compared with the route counterparts, as the semicircle, especially in the DI solution, or in the DI lactic acid, you could see clearly that additively manufacturing sample is behaving much better. So here is also the potential dynamic polarization data and also the equivalent circuit models. We have drawn based on the results I showed you in the last slide. Again, based on the corrosion current density trends for the SLM and ROT, also shows that SLM is behaving slightly better as compared to the ROT counterparts. And so finally here, I would like to conclude this project as well, that the microstructural examination of SLM Ti-6,4 revealed a relatively fine and higher concentration of beta content as compared to the ROT. And also XRD analysis confirmed the presence of higher beta phase in SLM as compared to ROT. Corrosion analysis shows a better corrosion resistance performance for SLM as compared to ROT. And finally, the ROT Ti-6,4 showed increased corrosion resistance only in this and this electrolyte here, which is artificial saliva plus lactic acid, and deionized water plus calcium chloride, and also deionized DI plus lactic acid here below 6 pH. And final conclusion is both SLM and ROT predicted significantly reduced corrosion resistance in artificial saliva and lactic acid and DI plus fluoride solution as compared to all the solutions we have studied. So with that being said, thank you for listening to me. And here is my email, haide1w@cmich.edu. So I would be very happy to answer any questions. So please feel free to write me. And thank you very much for listening. Have a great day. Bye.
Okay, thank you very much, Professor Dr. Wasem Haider, for your excellent presentation that give us new paradigm and knowledge. Maybe there are some questions for you from the all audience. If you have question for Professor Dr. Wasem Haider, you can email the email Professor Haider, and the email address in the chat box by committee. Okay. The next speaker, we continue. It's now my pleasure to introduce the next speaker. Is Associate Professor Dr. Intan Safinat binti Ismail. She is an Associate Professor from Department of Chemistry, Faculty of Science at University Putra, Malaysia. Her educational background, she get her Bachelor Science from Creighton University, USA, and received her Magister of Science from University Putra, Malaysia, complete her PhD and postdoctoral studies at Okayama University and Hoshi Medical University from Japan. She joined University Putra, Malaysia in 2005 and become head of Laboratory of Natural Products at the Institute of Bioscience in November 2011 until May 2017. Now, she is the head of Chemistry Department, Faculty of Science, University Putra, Malaysia. Within the period of her affiliation in the University Putra, Malaysia, he has published around 117 papers in reputed journals and about 95 presentations at conference, and being invited speaker, including a keynote speaker at international meeting. She is editor for Journal Natural Medicine and International Journal of Chemistry and Research and chairing a few important scientific meetings, including ASEAN Symposium and Medicinal Plants and Spice for 2021. Now, so as to start the second invited speaker, allow me to invite Associate Professor Dr. Intan Safinar binti Ismail to join with us and to present her presentation with the topic Current Development of Natural Product Research in Malaysia. It's such an honor to welcome you here in this event, Prof. Intan. To Prof. Intan Safinar binti Ismail, the time is yours, please.
Thank you very much for the kind introduction. Can you hear me?
Thank you, Prof.
Okay. I don't know whether I have to control my slide or not. Is anybody controlling my slide, or should I control my slide?
You want to committee to help your presentation, your slide?
Sure.
Yes, of course. Okay, your presentation is...
Okay.
Yes.
Thank you very much.
Okay, Prof. Intan, you have 30 minutes to present your presentation in this session, and after Professor Dr. Zaidi, we continue the discussion part with other speaker to answer the question.
Okay. Okay. Assalamualaikum warahmatullahi wabarakatuh, and good morning. Good morning. Okay, still morning. Thank you for the invitation, especially to Dr. Hollis and also the committee of ICAMs. I'm glad to be here this morning. But before I go on with my talk, I just would like to invite all to the ASOMS next year, which is ASOMS stands for Asian Symposium on Medicinal Plant and Spices. So it was supposed to be this year, but because of the COVID-19, we rescheduled it to next year. From 17th to 19th of August in Putrajaya. Next, please. Okay, so this is a title that's been given to me, so I don't have much choice. But I would like to just highlight that I'm not going to be talking the whole scenario of the current development of natural products in Malaysia, because it will be quite a long talk to speak on. So I'm going to be focusing on a small part of it, which is what we call omics, and how omics are being used in the development of natural products in Malaysia. Next, please. Okay. Of course, as we say in there, we are now facing this big problem, which is COVID-19. And without showing this, I think, and maybe probably somebody is asking why I'm showing this. This is related to whatever that my talk is about. So this is the world dashboard of the scenario of COVID-19, but specifically on the tracing. So this is the tracing ability of COVID-19 around the world. As you can see, the red colors actually referring to the countries that we could not be traced the case COVID-19 cases. So that is probably due to the technology, which is not that advanced. So when we're talking about advancement, we're not only talking about, advancement of natural products per se, but we are also talking about the technology of application in natural products. Next, please. So how are we combating the COVID-19? It's actually there are several techniques that are being proposed. And then one of the famous techniques is actually looking for the vaccine. Now under WHO coordination, there are about 169 candidates for the vaccine, but only 26 of them are now in the human trial phase. And there are several countries which come together under WHO trying to find the best vaccine for the COVID-19. Other than that, of course, there are also other technologies that have been proposed. For example, the breath testing system, which is being developed by Shimadzu Japan. And this is very interesting because University of Gajah Mada also down there, they develop what we call genomes. This is actually not as a vaccine, but this is to replace for a faster detection of COVID-19. Now we are using swab, and then we are using qPCR and PCR for the confirmation. We have the kit, but sometimes the kit give us negative or positive results, false positive or false negative results. So this breath testing as being developed here probably can replace the time-consuming or also cost-consuming of the swab method. Next, please. But not to forget, we are looking at the bigger scenario, which is COVID-19. But at the back of that COVID-19, there's also existence of other kind of diseases. So if you can see globally, there are infectious and non-infectious disease that we are still facing. They are not going anywhere actually. For example, the mosquito-borne diseases. Before we are familiar with dengue and malaria, but now there are so many kinds of variety that came out from these mosquito-borne diseases, like for example Zika and GE. And other than that, that is infectious. We are also facing the drug resistance from our microorganisms, and that is infectious diseases. But how about the non-infectious? Each of the country in the world, as you can see there, we are facing at least one or more of the non-infectious disease. And the one that I'm pinpointing down there is the obesity. Malaysia is one of the having the highest number of obesity cases in Southeast Asia. And half of the 5-year-old children that is facing obesity live in Asia. So that is a very big problem. So other than COVID-19, there are also other problems that we are still facing. Next, please. Okay, so we are trying to get the drugs and also candidates that can combat this disease, either the new one, COVID-19, or the one that been existing since the old time. So this is the common scenario or common flow chart that we can see when we do the drug discovery and development. So most of the time we're going to be choosing a disease, and then we have to find the drug. Finding the drug is like finding a needle in haystack. So it's not going to be simple work. So when we get a drug, the lead compound, we have to optimize the lead compound so that we can hit or go to the clinical trial before we hit the market. But the main problem is, even when after spending years and money and time and of course energy, we could hit the market, and then after hitting the market, some of the drugs will be facing with the withdrawal. Why? Because, can we go to the next, please? Because if you see here, probably we are not looking at the right candidates, because in the pool of the drugs that we got, there are some which is called PINs. PINs are actually referring to pan-assay interference compounds or colloidal aggregates. And when the PINs come from natural products or natural resources, they are called IMs, which is the invalid metabolic panaceas. So these particular compounds is actually giving the false positive. And in 2017, there are about 60 FDA approved drug have been withdrawn from the market because of their characteristics of being as IMS. And 25 of them is actually from the natural products. Next, please. So when we are facing this problem now, we are facing a lot of problems for in the global diseases, including the COVID-19. We are also facing the technique that we're using. Now we are using the reductionism approach, whereby we choose a target and then we find the drug to combat the target. Another thing that we are facing, a problem that we are facing is the situation, the global situation, the changes, which is the biodiversity with depletion. Biodiversity now is depleting more than what we can replenish. And natural products resources are also depleting. We are also talking about some drugs which originated from the source of, for example, the synthetic drugs originated from the oil supply, also depleting. And we are also having the growing population. Now we are having 7 billion population in the world in 2020. But in another 30 years, they're going to be another 3 billion addition, which is extrapolated going to be 10 billion in 2015. So looking at this problem, we are facing with a lot of people, more people, increment of population. But on the other hand, we are also facing the depletion of biodiversity and also the natural resources. Next, please. So, what I'm going to show you now is the omics. Just now, I showed you the flow chart or the mechanism, the flow, the method that being used. The way that we call reductionist, whereby we choose a drug and then we didn't look at holistically. But omics actually giving you a chance of looking at the system globally or holistically. So you can see here, if we choose an experimental system like a human, we can use the samples from the human, from several parts or organs, or even, for example, biofluids, and then we can see it based on either genomics, looking at the gene, or transcriptomics, looking at the transcripts, or proteomics, looking at proteins, and the last one, which is the terminal of the omics system, which is metabolomics. But if we conclude everything, we relate from genomics until metabolomics, what we can get is we can get a system biology. And this system biology can give you a network integrated data or protein-protein interactions, as I shown here. Okay, a part of the omics is metabolomics. Next, please. So metabolomics is actually a part of the omics world, which is a non-biased identification and also quantification of metabolites in a biological system. So the biological system can be a cell, can be a tissue, and also can be an organism, so like a human. So we can see the phenotypic expression when there is a xenobiotics or effect from outside. Next, please. But when looking and applying omics, we cannot forget about the 5 pillars or 5 rules that I would like to introduce also. So these 5 pillars actually based on what abbreviated as QSECA. QSECA is actually referring to quality, safety, efficacy, consistency, and also availability. There are so many natural product chemists out there that look at a candidate for certain disease, but forget about the quality and forget about safety of the natural products, and they jump straight into looking into the efficacy of the natural products or natural resource. But actually, before jumping into the efficacy, it is very important for us to know the quality of the materials that we use and also the safety of the material. After we get the efficacy, we also have to understand whether the efficacy is consistent also or not. And the main problem now, because I relate to the natural resources, availability of the material that we use probably is not going to be there. Next, please. I'm going to be focusing now on my study specifically on Clinicanthus nutans. Actually, Clinicanthus is belalai gajah or Sabah snake grass. It's very widely distributed in Thailand, Indonesia, and Malaysia. And it's being used as a traditional medicinal plants. Next. And the use of this plant is actually related to anti-inflammation. So what we have done is we focus on neuroinflammation in the brain of rats. So we use the rats as our model and looking at the plants, the Clinicanthus nutans. We started with the plant harvest, which is related to the quality of the plant. We use only one plantation so that we will not get variation in metabolites. And then we look at the extraction method, and then we screen the activity by using the in vitro study before going into the rat. So we specify our technique so that we don't waste time, too much time. And instead of using only one platform to analyze the data, we use two platforms in integration and complementary with each other, which are Proton NMR and also LCMS. But I'm going to be focusing on Proton NMR for this talk because I'm not going to be spending a lot of time talking about the data. And of course, we also relate it to the biological activity, firstly with behavioral of the rats after treatment and before treatment, and looking at until the gene expression in microarray, and then we simplify the data observation through multivariate data analysis to identify the biomarker which is in the rats, which is giving the different expression between the healthy rat and also the treatment after induction of neuroinflammation. Next, please. Okay, this is an example of quality. When we talk about quality, we are talking about the treatment of the plant extract itself, the plant material. So before we go into the extraction, we have to also understand how the plant being treated, which is the drying method. I didn't show it, I'm not showing it here, but the drying method is also important. So this is the example of the solvents being used, 3 of them, which are aqueous, 50% ethanol, and ethanol 100%. Why we are using ethanol? Because ethanol is the most friendliest industrial solvent, organic solvent. As you can see there, even using these different three solvents, we can see that there are different expression of metabolite being extracted. Aqueous can give you a range of metabolites, but 50% ethanol and ethanol give a very high sugar content. Next, please. So from that particular spectra, 3 of them, we extracted and identified some of the compounds in here, which are 44 of them, and we combine them based on the classes which are primary and secondary metabolites. Primary including the intermediates, and if you can see there are many of them, many amino acids and also secondary metabolites of phenolic and phenolic groups. Next, please. So after doing, I have gone through the in vitro assay, and then we pinpoint which particular solvent is good to be used. We went on into in vivo rat model. So for this in vivo rat model, what we have done is we relate the behavior study with a cytokine antibody and also the serum from the blood of the rats. Next, please. So this is the experimental design that we use. We use the Sprague-Dawley rats, and we divide it into groups with the positive control that being used, dextromethorphan, and also the vehicle only. And then we see it for 1 month, whereby we acclimatize it for 1 week before we induce in through geotasic brain operation, and then we induce it with lipopolysaccharide LPS for neuroinflammation, and after that we treat it with Clinacanthus newton extract, water extract. Next, please. So we withdraw the blood, and we separated the serum from the blood, and from there we submitted to the NMR, whereby we can see here there are several groups that I showed you just now, including the positive control, the induction of the LPS, and also the treatment of Clinacanthus at several doses. And you can see here, we use 250, 500, and 1,000 milligram per kilogram body weight of Clinacanthus to the rats. And we look at after the 14 days of treatment, where we sacrificed the rats and got the serum. So you can see here, it is quite difficult to see by the naked eyes, even though you can see some of the chemical shift giving different signals. So what we have done is, we translated it into multivariate data analysis. Next, please. Into the plot like this, which is called principal component analysis plot, score plot. As you can see there, the groups can be clustered into 2 clusters. One cluster is at the left side, which is negative quadrant, which is group the normal and also the normal plus 500 milligram per kilogram of Clinacanthus, showing that you taking 500 milligram per kilogram of Clinacanthus will not give you negative effects. And when we induce with lipopolysaccharide LPS, we can see that they are being induced and they're clustered at the positive side of the quadrant, and they share the same metabolites being neuroinflamed. And you can see at the B plot there are 3 clusters now separated the groups. And you can see that dextromethorphan, which is the positive control, now being grouped similarly with normal and normal 500. As I said, normal is actually normal plus only water alibidem. So the one that is at the upper side of positive quadrant is actually the LPS, the induction, and also the one that is being treated with 250 milligram per kilogram Clinacanthus. So meaning that 250 milligram per kilogram is not giving a very good effect. So we can see that the cluster of 500 and 1,000 is giving a little bit different from the LPS induction. Next, please. So this is the behavior that we relate to the plot just now, and then we whereby we see 29 parameters, and from these 29 parameters, we classified it into a few simple parameters under the same category, which is psychomotor, speed, and anxiety. Next, please. As you can see here, this is the behavior in a box arena. We call it arena. When we induce it with LPS, we can see that the movement of the rats is only at the peripheral of the arena, when compared to the normal one, whereby they cross the middle center a lot. And we can see the differences when it's being treated with 250 and other treatments. And we extrapolated the movement into some of the parameters, and we put it in the bar chart, as you can see there. Next, please. And of course, it's very difficult to see the bar chart, and even in the arena, what we have done is we again put it in a multivariate data analysis, whereby we use orthogonal partially square discriminant analysis, considering all the parameters, but simplified it so that we can see. And we know that dextromethorphan, the positive control, and also 500 milligram kilogram per kilogram is different from the normal rat and also the LPS induction. And we can see there are some of the positive effect is there. And we also extrapolated this validated regression model down there. As you can see, there is the regression validated model, whereby we can use it as a predictive model for the future use of the Clinicanthus Newton samples without going into the in vivo assay. Next, please. So this is how we choose some of the compounds that can be related to the behavior and also can be related to the serum. So this is based on the variable important projection, whereby we selected only the significant compounds that related to the changes of the treatment. Next, please. And from there, we put a map, and then we map out all the VIP values, high VIP values of the compounds, and then we see between the groups the changes in the map, which is at one glance you can see the difference from where the normal and the treated can be divided into groups by hierarchical component analysis. Next, please. We relate the compounds that we nominated by VIP into by using MetaboAnalysis, which is an online application software, whereby we choose the mechanism or pathway that being impacted a lot by the metabolites that being identified. Next, please. And this is the brain tissue. So we look at the serum, and also we can look at the tissue. And then the same way that we look at the serum, we went on using NMR, and then we simplify it by looking into multivariate data analysis. Next, please. So before we look at we put it into multivariate analysis, we relate it with cytokines, whereby we express it by microarray, and then we got the cytokine expression. And these are the cytokines that we use listed there from B, C to F, and then based on these cytokines and chemokines, one of them are chemokines, MCP1. We put it in the bar chart so that we can see the expression differences between the groups. Next, please. And then we put it into multivariate analysis so that we can see the differences or the movement of the changes in the groups. Next, please. So these are the tables that given the expression of the cytokines. I'm sorry, I'm going to be speeding, ending this presentation. Next, please. So from there, we understand what kind of, in the organism, what part have been impacted or perturbed much in the system of the rat model, which is immune system and also PKG mediated neuroprotective protection. Next, please. So this again, we use the tissue to go into the pathway and selected the pathway that being perturbed. Next, please. So this conclusion, so meaning that instead of using the reductionist, looking at only one window, now we are opening the window wider so that we can extrapolate the reaction or the treatment of Clinicanthus into some parts and related that particular results into one system of holistic view. With that, I thank you for your attention. Thank you very much.
Okay, thank you very much, Professor Intan, to share your great presentation. So we can get the new knowledge, and the topic of natural products is very interesting because some research now lead to traditional medicine, use plant and other materials. Okay, Professor Intan, We will talk discussion about question, but now we continue the last speaker. Is okay?
Okay.
Okay.
Thank you.
Thank you, Prof. Okay, it's now my pleasure to introduce the last invited speaker today, Dr. Ruzaidi Azli Muhammad Mokhtar. I'm sorry. Dr. Ruzaidi is a lecturer at Biotechnology Research Institute, University Malaysia Sabah. And he received his Bachelor of Science major in Applied Biology Science from Tokyo University of Agriculture and Technology, Putra, Malaysia. And he get his PhD degree in Physiology, Monash University, Victoria, Australia. And he has been appointed as Deputy Director of Research and Development Biotechnology Research Institute in August 2011. And had various other positions along the length of the service with University Malaysia Sabah. He was involved in diabetes research since 2000 when he pursued his research mainly on diabetes. He also obtained several research grants from the Ministry of Higher Education and Ministry of Science, Technology and Innovation, Malaysia. As a result of his studies, he has received several awards at the university, national and international level. His findings were published in several international academic journals. And now, so as to start the last invited speaker, allow me to invite Dr. Ruzaidi Azli Mokhtar to join with us and to present her presentation with the topic anti-tuberculosis and anti-dengue activity of selective mangrove plants extract from Sabah, Malaysia. It's such an honor to welcome you here in this event, Dr. Ruzaidi. To Dr. Ruzaidi, okay, the time is yours, please. Yes. I can hear your voice. Okay.
Okay.
Good morning.
Thank you, Mrs. Chairman, for the introduction. First of all, I would like to thank the organizing committee for inviting me to present in this conference. I'm quite excited to share some of our research that's been done with Swiss Germany University with the Dr. Collis on anti-tuberculosis and anti-dengue on mangrove plants. Should I share my screen or?
We can help you. Okay.
Yeah. Okay. So as I informed previously, this project is a collaboration with the Swiss German University on the anti-tuberculosis and anti-dengue activity. So for my parts, for this presentation, I just focus more to mangrove plant from Sabah. So definitely we will screen plants from Indonesia as well after this. Okay, next slide, please. Okay, why we choose mangrove in this project? Because of as we know that mangrove live at a very harsh and stressful condition, which is have a high salinity and so on. And then a lot of study is shown that these mangrove produce a lot of bioactive compounds that maybe is beneficial for range of therapeutic significance. Yeah. So next. Okay. I think Dr. Collis already give a talk presentation about the mangrove distribution in the world. I think Southeast Asia is the largest area of mangrove in the world, which is I think more than 30%, and Indonesia I think is the largest in the Southeast Asia, and I think Malaysia is second. So what I want to focus for my presentation is more to Malaysia's scenario, which is in Malaysia, Sabah is the largest area of mangrove, followed by Sarawak and Peninsula. So 60% of mangrove area is come from Sabah. So next. So because of that, that's why we choose this mangrove, because there's none research is done on the mangrove in Sabah, and plus several research has proved that these mangrove have antibacterial and also anti-tuberculosis activity. Next. And also some studies shows that smoke from burn extract mangrove kill mosquito larvae, which is Aedes that can cause dengue. And that's why we select this mangrove to study about its anti-TB and also dengue. So consider that this is serious issue in this region, especially in Southeast Asia. I think TB also is a main problem in Indonesia, and also main concern in Malaysia, especially in Sabah. Okay, next. Okay, a little bit statistic about tuberculosis in Malaysia. Okay, as I said, Sabah is, because I'm based in Sabah, University of Malaysia Sabah. Sabah is one of the highest TB cases after Selangor. But if we compare the population, Selangor of course have a higher population than Sabah. In terms of percentage, Sabah is the most high incidence of TB in Malaysia. So for your information, maybe some of you don't know about TB. It's caused by the Mycobacterium tuberculosis. It's primary affects the lung, and about the, yes, of course, there's a drugs to treat this TB, but the multi-drug resistance is always being problems in the current drugs treatment now. So that's why we propose the alternative solution to treat this TB. So next. Okay. Again, dengue also is a quite common problem. I think both in Indonesia and also Malaysia. In Malaysia, is increased almost every year. So last year is increased to 130,000, more than 130,000 cases, and caused 162 deaths in Malaysia. So at the moment, of course, there's no specific treatment for dengue. And then of course, our intention here is to search for the alternative treatment for this virus. Next. So as I said, I just for this project, the main purposes of this project is to evaluate the anti-TB activity of the selected mangrove plant extract, and also to evaluate anti-dengue of the selected mangrove plant extracts. Next. Okay, this is the overall methodology. Yeah, we collect. We just able to collect the sample only from the one sample site because of the limitation of the budget. Of course, in future we're planning to do sampling all around Sabah, especially the Lahad Datu area and also Tawau area. I think that area is a very high. A lot of mangrove species over there, but for now, I just present the sampling site near Tuaran, which is near our campus. It's only 40 minutes from our campus. So after we collect the sample, of course, we do the extraction. So we use 4 different solvents, which is aqueous, ethanol, and all ethyl acetate, and also hexane. of course, we measure the total phenolic and antioxidant activity, and after that, we screen this sample on the anti-TB and also anti-dengue. Next. Okay, this is the sampling site. So I think maybe some of you don't know where is Sabah. Sabah is located at the Borneo Island. It's, if Indonesia is the Kalimantan, so we just north of the Kalimantan. Okay, so next, basically we're there, and then that's our sampling site. As I said, it's only about 30 to 40 minutes from our campus. Next. This is the bigger map for that. It's quite near, and then we managed to collect about 6 species in Sulaman Lake forest, mangrove forest. So next. So this is the sample area, sample sites. I think it's quite nice view here. I think Dr. Collins also been there. I think last 2 years. To see where we collect all our samples. Next. And then this is the samples that we able to collect at Sulaman Wetland Tuaran. So basically it's comprised of 6 species, and then in total is 13, I think 13 parts of samples. Next. It consists of 13 different parts, and we extract it with the 4 different solvents. So in total, we have about 52 samples to screen for anti-TB and also anti-dengue. Next. Okay, this is some picture. Some of my research assistant and students doing the collection, and also do some preparation before extract the plant. Next. Okay, I'm not going detail on how to do the extraction. Go directly to the result of the anti-TB and also anti-dengue. So for information, this project is not that new. It started 2018, but until now, we managed to finish all the anti-TB screening. But unfortunately for anti-dengue, we already optimized all the methods. But I don't know whether you all know or not. Currently, Sabah is under lockdown. We cannot go to the lab. Basically, we still didn't manage to finish the anti-dengue screening. But once we can get back to the lab, I think you will finish it by 2 weeks. So because all is ready, but we just waiting to go back to the lab to finish our study. But I will present what's our preliminary data for this project. So next. Okay, so for your information, for anti-TB, we do it in our BSL3 lab, a biosafety laboratory lab. So we have our BSL3 lab here. So it's quite fortunate for us to have it. This is how they do it in the lab. Quite. They need to dress properly in the lab. Okay, next. This is how they prepare the strain. Our strain, we use the Sabah strain. It's not the standard strain. And then next. Okay, this is how the bacteria looks in the tube. If you can see the white breadcrumb like structure, that's a TB. Okay, so next. Okay, so for the screening, we use the Resazurine microtiter assay. So basically, this is how my students arrange the samples for the test. So basically, one 96 plate, they only can screen for 3 samples. So okay, yeah, of course we need to have a positive control, standard, and negative controls. And then next. Okay, once they put it in the 96 well, we will put the extract, the mangrove extract, into the well, and then incubate for 5 days, and after that they put the resazurine dye, and then incubate another for 2 days, and then they determine the MIC. And so the whole is quite long, even though the TB bacteria grow very, very slow. I think the whole process is normally is 2 weeks to get the results. Okay, next. Okay, this is how the plates looks like. So blue color indicates the absence of the TB, and the pink indicates the presence of TB. So basically, we will choose the MIC to see which mangrove extract have the higher inhibition properties. Next. Okay, this is I think the final result. We've already done all the screening for the Sabah mangrove extract. So as I said, we have total of 52 samples. So next, so the highest anti-TB activity is in the Bruguiera gymnorrhiza leaf, and also the Cereoptaga leaf, and using the ethyl acetate as a solvent. So as you can see here, the ethyl acetate gives a very high anti-mycobacterial activity compared to the other solvent. So in general, we can see in the next graph, yeah, basically ethyl acetate and also the hexane have better antibacterial activities compared to the aqueous extract. Okay, next. Okay, to summarize the finding for this one, the range of the MIC for the mycobacterial activity is from 0.04 to 5 milligram per mil. And then the best one, as I said, is in the ethyl acetate extract of Bruguiera gymnorrhiza and also Cereoptaga leaves, which the MIC value is 0.04 milligram per mil. So we do some comparison with other studies. As you can see, the other studies, the MIC is quite high compared to our study. Even our lowest MIC value is 5 milligram, also consider is higher than the other studies. So it's good indicator that all these extract have very good, I can say, a very good anti-tuberculosis activity. So in future, we will use this extract for the analysis. So especially for this project, we will look at the compounds maybe that responsible for these activities. Okay, next. So we go to anti-dengue viral activity. As I said, unfortunately, it's still not complete, but I will present also what we do in this study. So next. So yeah, this is I think the hardest part for this study because my student scientists take around 6 months to optimize this method. And then finally, she get the 80% purity of protein from. We use the NS2B and NS3 protein here. This is a specific protein for dengue, to see whether mangrove extract can inhibit this protein. Okay, I have several preliminary result for this one is just for the optimization of this method. So next, is how my students do the optimization. This one is they optimize the incubation temperature. I think yeah, this one is computer temperature. I cannot see the at the bottom. I think they select this one. Yeah, the bottom right, which is they incubate it overnight for 20 degrees. Yeah? That's give the better band compared to other temperature. Next. Yeah, next. Okay, the protein is at about 38 kilodalton. So this is after the purification, and then finally we managed to get more than 80% purified protein for the study, and now I think it's ready to be used to screen the mangrove extract. But yeah, unfortunately we cannot do that right now. It's all the lab is closed. We hope that we can do it soon, so that we can present it here. Okay, next. Okay, after this, we will test this with the compound with the 52 samples, and then this is the method, and we will determine the IC50 and measure with the 3D duplicate. This is how we will do it. After this, next. And then at the beginning I just mentioned that we also measure the total phenolic content of all these mangrove extract, but unfortunately I just can present you the aqueous and ethanolic extract, because we lack of sample for ethyl acetate and also the hexane, because it's quite hard to get the sample. Next. Yeah, we use the Folin Kokatu method for total phenolic content. So next, as you can see, next the highest total phenolic content is in the Streptococcus leaf. Next, the next one is in the Rhizophora mucronata leaf, and the third one is the Bergera gymnoriza root. So this is the best three highest total phenolic for our samples, but actually we've done several samples on ethyl acetate and also hexane, but I think the value is not that high, so we can say that the total phenolic and the anti-TB activity is not correlate at all. Okay, but next is we also measure the antioxidant activity using the DPPH method. Next, and as the results, yes, ethanolic extract give the higher inhibition compared to aqueous, and then the highest is in the leaf and the Streptococcus leaf, and also Rhizophora mucronata leaf. Yeah? So basically, it's most of the sample, the leaf contain a higher antioxidant and also the total phenolic content. But again, as I said, we cannot relate it with the anti-TB activity. So most probably the anti-TB activity is not come from this the phenolic or phenolic compound. Next. Yeah. Okay. We're planning to do see what is the bioactive compound responsible for the anti-TB activity, but as I said, this one also is a quite preliminary study. We just run it one sample. We just managed to run the ethanolic extract of Bergera gymnoriza leaf, LCMS, And then next, we got some results that indicate that it contain several compound that reported have the antibacterial activities. Next, I think is in the next table. Next, please. Okay, this is the compound that we managed to identify in the Bruguiera gymnorrhiza leaf, which is a benzoic acid. It's previously reported as antimicrobial and also antimicrobacterial. Even we can detect also ethyl benzoate. It's an antioxidant and anti-inflammatory. Ethyl vanillin is antibacterial and antimicrobacterial activities, and so on. As I said, this is a very preliminary study. We definitely will go to analyze especially the ethyl acetate extract, which shows the highest activity for TB, and also we will analyze the extract that show the highest for anti-TB. So next. Okay, for future work, as I said, we go for phytochemical analysis. Especially for the extract that present the highest inhibition, both for anti-TB or anti-dengue activity. Even our planning is to do fraction, and even if we can get the compound, the single compound that responsible for this activity. So also next, I think mostly we've done with go back to the previous slide. Basically, we've done with the Sabah samples. But in this project, we one of the objectives is to compare with the sample from Indonesia. So maybe Dr. Collis will give some samples for us to compare whether have a similar activity or not. So next, we also proceed with the immunology study. One of our my PhD students will proceed with immunology study to see whether this mangrove extract can affect our immune system. Okay, next. Okay, last but not least, I would like to acknowledge the people involved in this project. First is Tamar is my PhD student. The second one is quite hard for me to pronounce. But I call her Ain. It's my master students. I think it's involved a lot in the screening processes. And my colleagues, Dr. Zarina, Dr. Nakira, Dr. Chayo, and also our collaborators from Swiss German University, Dr. Collis, Harris Sutanto, and also last but not least, the staff from Sulaman Wetland, Mr. Hafiz and Ms. Mary. So this research is funded by UMS, it's a collaboration research grant, and also research grant from SGU. So with that, I thank you.
Okay, thank you very much for Dr. Zaidi Azli Mokhtar for your very interesting and innovative presentation about the mangrove plants from Sabah, Malaysia. Okay, now we have to continue the next session. This is discussion session. So for Professor Intan, still with us, Prof. Please, you can join again in panelist discussion. Okay, and for our audience and international conference on agromedicine and medical science, we have just listened to the presentation from Professor Wasem, Dr. Zaidi, and Professor Intan. Now, I still remind you again, if you still have a question and comments, do not hesitate, just write in Q&A box. Okay, now we continue with discussion. Maybe for Professor Intan and Dr. Zaidi, we see the Q&A box. Okay, this is there's some question from the audience. maybe, but due to the limitation of the time, maybe I will just pick some of them because we have time for discussion about 10 minutes. Okay. Okay, now I will read the first question. The first question is from Mr. Samswar from University Tulang Bawang, Lampung. Okay, Mr. Samswar would like to ask for Professor Dr. Intan Sapinar. There is a problem, Prof, for the development of herbal medicine product. The price of herbal medicine relative expensive compared to modern medicine. What is the solution from you, Prof? Maybe you can answer, please.
Sure. This is my opinion, actually, looking at the scenario now. When we got into drugs, to go to the drug level to get a one entity drug is expensive. It's not comparable to the herbal drugs, actually. We didn't see the cost of producing a drug, because after we got a candidate, what is the best practice is they will synthesize, and then they can produce in high volume. Okay, so from the small scale, they can be upscaled by synthetic drugs. But with herbal medicine, you cannot do that. How can you synthesize a herbal medicine? So that's why the cost will be not going to be decreased. It's going to be the same. So if you're starting with a herbal drug preparation, it will be the same throughout the time, because you cannot have the second choice of synthesizing the products. So that is the main thing. But the problem is people tend to forget about the quality of the herbal products. They just put it in a capsule or in a sachet, and then they just nominate it as a good treatment for something. But how about the quality of the products that being produced? So I think that is the one that have to be think about, to be thought about, and then to be really with good collaboration with the government and also agencies under the government or your government, and also any government, to look into this so that the efficacy is there. Do you understand? Because people can sell anything in a capsule, and people can sell anything in the sachet as tea decoction, but we don't know whether it is good or not, and people will buy. That is the problem of herbal medicines, expensive or not. Okay, that is my answer for that. Thank you.
Okay, thank you, Professor Intan. Well, this is satisfied from answer, yeah, Prof. Okay, I think the answer is quite clear. Okay, maybe we next the second question. Okay, this is from Dr. Handi Siono. The question for Dr. Ruzaidi. Okay, in comparison, which is the poor and good antimicrobial activity from the leaf, stem, or root of mangrove, Doc? Please, Dr. Ruzaidi.
Okay, thank you for the question. So for your information, I'm not doing any antibacterial activities. I'm more focused to anti-TB, which is antimicrobacterial activities. If you want to see the in general antibacterial activity, I think the best person can answer is Dr. Collis Auda. I think he's doing a lot of research on antibacterial activities of mangrove plant. But in terms of for anti-TB, what we can see, we cannot see any tendency in terms of different parts of plants. I cannot say that the leaf is have a highest anti-mycobacterium activity or not. But what we can see here is the tendency of the different solvent. As I said just now, the ethyl extract give the highest anti-TB properties compared to the other solvent. That's what we can see the tendency. But in terms of different parts, we didn't see any tendency which part is the best or not. That's my answer.
Okay, for the answer, maybe Dr. Hollis, yeah. Okay, but I want to continue for Dr. Zaidi. You research for mangroves with to TB and dengue. Whether mangroves are better dealing with TB, tuberculosis, or dengue fever, Dr. Zaidi.
Sorry, I cannot catch. What's your last question?
The last question is whether mangroves are better at dealing with tuberculosis or dengue fever, the effect.
Or dengue fever?
Yes.
Yeah. For now, I can say it's dealing better in TB because we have the results. For the dengue, we still don't have any result,
okay.
To compare with. So I cannot say anything for anti-dengue activity. Yeah.
Okay, because you have the future work, maybe I see phytochemical analysis of the extract that represent high inhibition of anti-TB and anti-dengue activity, maybe.
Yeah.
For your future work, okay.
Yeah, for future, yes, yeah.
Yes, okay. Thank you, Dr. Zaidi. Okay. The next question, there is from. Oh, there is no names, but this is for Professor Intan. How will what it is? How ethanol will affect our body, and what kind of activity that will increase the risk of ethanol abuse, and what can we do for recover from the illness? Maybe this is a question for the effect of ethanol, Professor Intan.
Okay. So actually, we extracted the materials in ethanol, but we are not going to be having the ethanol in our material. Do you understand? I hope you understand that.
Okay, yes.
Because we extracted and then we dry. So most of the time, what we will be doing is we go until the step of freeze drying, making sure that the ethanol is not there. Also, in the industrial stage, what they will do is the industry will take the extract, and then they will be using the oven to heat it up so that they will dry up the ethanol off, because 70% ethanol will be alcohol, and
okay.
everybody will be drunk.
Yes. Maybe it's different because we extract the ethanol. Okay. Okay. Thank you, Professor Intan. The next question for Dr. Zaidi. This is the question is okay. Which part of mangrove is more easy to process to extract, Dr. Zaidi?
Which part of?
Yes, the parts of mangrove. Maybe there is the leaf and root. Maybe the means of the question.
Yeah, it's clearly. So to me, it's the leaf, because the roots and the stem is quite hard sometimes. It's also hard to dry. It take time. Yeah, and then I can say it's the easiest parts in mangrove is the leaves.
Okay, thank you very much for Dr. Zaidi. Okay, this is some question from anonymous again, but this is for Professor Intan. In your research, this is use dextromethorphan administration function in your research. What is function maybe?
Dextromethorphan is actually a drug used for neuroinflammation. So this is a positive control that we use to compare. So every time we do bioassay, in any bioassay, we need a control so that we can compare with. For example, the most famous now that people, I think some of the people around the world, they take curcumin for anti-inflammation against COVID-19. I'm not saying that this is good, but that is an example of positive control. People have known the drug as a good drug against neuroinflammation, so that's why I use it so that I can compare the activity of the plant against the positive control. Thank you.
Okay, thank you, Professor Intan. It's clear quite. There is one question for Professor Intan before we the last session. It's from 30 chemical marker for anti-neuroinflammation in vitro study were identified. Which marker give more important effect?
Okay, in the slides, if you can see that I showed you VIP, variable importance projection. So from this, actually I identified 44 compounds. 44.
Okay, 44. Okay.
44 compounds. It's okay, no problem. From that 44 compounds, we nominated only about 10 of them, which are giving a very significant effect. So we cannot nominate all 44 because it's very difficult to see. So from this 10, I didn't share with you guys because I don't want to spend too much time. But actually from these 10 compounds that we nominated as significant compounds, we took it and then we did a full change, meaning that we have a quantitative, not only qualitative. So when doing bioassay, we cannot just look at qualitatively because it will give you a very surface results. So we have to go deeper into quantitative. So we took the 10 and then we did the quantitative measurement so that we know the full change. Full change meaning that, for example, glucose in glucose in the body, it will increase because of the inflammation. So how much they increase? Sometimes you can see that it can increase 3-fold, 2-fold. So from there, we understand which one is important. So if you can look back at my presentation, the 10 that I took them and then put it into the heat map, they are the ones that are important biomarkers. Thank you.
Okay, thank you very much. That's interesting answer from Professor Intan. Okay, for our speaker, I think the question are enough for us now, and due to the limitation of the time, all questions have been answered clearly by all invited speaker. Maybe if there are still many or some unanswered question, the audience can by email Prof. Intan or Dr. Ruzaidi?
Yes, please. You can email me.
Thank you, Prof.
Thank you.
Thank you to our questioner. Next, for all our great invite speaker, for your L certificate, we will send the certificate to your email by committee. Finally, we came to the last session. Thank you very much for our honorable speaker, Professor Dr. Wasem Haider, Dr. Ruzaidi Azli Mokhtar, Professor Dr. Intan Safina Binti Ismail for the very exciting and innovative presentation that improvement our knowledge in medical science and technological in agromedicine is a specialty. And thank you for spend your time for us, especially for our speaker. And also thank you to all audience for your participation until now. Hopefully the information that we have shared today would be useful for all of us. We look forward to seeing you at the next event. Thank you for your attention. Okay. Thank you for Dr. Zaidi and Professor Intan. Maybe okay for Professor Intan. Yeah, bye bye. We can see in 2021 for your meeting. Okay, in
Inshallah.
Malaysia. Okay.
Yes, please. Thank
Okay.
you. Bye bye.
Bye bye.
Bye.
Well, that's wrap up this session. I return to the MC for the next session. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for all of the incredible speakers and also our moderator, Dr. Rani Himayani.
So we are already at the end of the first session. Thank you for Mr. Khalis Abdul Rahim Auda, PhD, as our keynote speaker, Professor Dr. Wasim Haidir, Dr. Zaidi Azli Mokhtar, and Dr. Intan Safinar binti Ismail as our invited speaker.
And we would like to thank Dr. Diana Mayasari, SKET MKK, and Dr. Rani Himayani, SPM, as our moderators, and also thank you for all of the participants.
Thank you for all the participants. We apologize for shortcoming right now. This is the end of the webinar session of the first day. Don't forget to all participants that we still have second day of this seminar sessions. Tomorrow we will start at 8 a.m. GMT 7.
Next, it's time for break, and to the oral presenter, we will continue to an oral presentations at 1 p.m. in different Zoom meeting. The email has been sent. Please check your email for the further information. Good afternoon, everybody. Wassalamualaikum warahmatullahi wabarakatuh.